Okay. Ma'am, the screen is seen.
Yes, you're visible.
Okay. Good morning to everyone. Today I represent my research work, and my topic is enhancement of azodie by remediation using activated biochar in a bioreactor. The first is abstract. The synthetic azodie waste from industrial poses serious environmental threats, especially the wastewater bodies. This study evaluates the use of Iconia crispus. Biochar is immobilized packing and material in a continuous packed bed bioreactor for the biodegradation of reactive orange. Dye using mixing microbial culture isolate from textile effluent. The results show that dye removal increased gradually and stabilized around the 10th day operation and achievement maximum removal of 65% at initial dye concentration is 500 milligram per liter. And the biodegradation was confirmed through UV visible spectrophotometer and FTR analysis. The result demonstrated that Iconia crespus biochar is an effective, eco-friendly, and cost-packing material for dye biodegradation and may also be used as a adsorbent and biofertilizer. अगर हम बात करें इंट्रोडक्शन की तो ड्यू टू इंटरडाइजेशन एग्रीकल्चर ह्यूमन एक्टिविटी कंजप्शन ऑफ डाई these are the these has been increased the polluted in the water body. The effluent discharge from this activity contaminate the water bodies, especially containing dye. The effluent from dye processing industries are highly colored, high BOD, COD content, and these dye prevent the penetration of sunlight, and it is dangerous for ecosystem. अगर डाई जो है वो सनलाइट के पेनिट्रेशन को रोकती है तो जो वाटर बॉडी है उसमें जो एक्वेटिक एनिमल्स और प्लांट है उनके लिए वो हार्मफुल है, हैजार्ड है. नेचुरल डाई आर नॉन बायोडिग्रेडेबल. जो डाई होती है उसको डिग्रेड नहीं किया जा सकता है. द डाई रेस्पॉन्सिबल फॉर बायोटॉक्सिसिटी, म्यूटाजेनिक एंड कार्सिनोजेनिक इफेक्ट, एंड मोस्ट ऑफ द डाई कैन कॉज एलर्जी, डर्मेटाइटिस और स्किन इरिटेशन. The next is synthetic dye like reactive orange and in textile effluent are persistent, toxic और कार्सिनोजन. डाई जो होती है वो कार्सिनोजेनिक होती है. हमने इसमें क्या फोकस किया है, पैक्ड बेड बायोरिएक्टर बनाया है जो एक लो कॉस्ट है. और जो आइकोनिया प्लांट होता है जिसे हम वाटर हाइसिन कहते हैं उसका हमने एक बायोचार बना के उसे यूज किया गया है डाई को रिमूव करने के लिए. प्रोसेस की बात करें तो रिसर्च मेथोडोलॉजी की बात करेंगे तो हमने बैक्टीरिया को आइसोलेट किया है डिफरेंट टेक्निक से. सीरियल डाइल्यूशन, स्प्रेडिंग, स्ट्रिकिंग एंड इनोकुलेट द बैक्टीरिया इन अ ब्रोथ मीडियम. उसके बाद सेकंड स्टेप की बात करें तो सिलेक्शन ऑफ इफिशिएंट बैक्टीरियल स्पीशीज. हमने बैक्टीरियल स्पीशीज को सेलेक्ट किया है उस वेस्ट से. उसके बाद हमने आइकोनिया जो प्लांट है जो कि खुद एक हैजार्ड है वाटर बॉडी के लिए, तो हमने उसका पायरोलाइसिस किया. पायरोलाइसिस के बाद में हमने बायोचार प्रिपेयर किया और इमोबिलाइज किया माइक्रोब को डाई में एंड फिर बायोरिएक्टर सेटअप किया एंड एनालिटिक मेथड यूज किया. फिर हम बात करें आइसोलेशन की टेक्निक्स की बात करें तो सीरियल डाइल्यूशन, स्प्रेडिंग एंड स्ट्रिकिंग मेथड हमने यूज किए. दिस इज सीरियल डाइल्यूशन. Serial dilution is a common technique used in many isolate procedure. A small amount of solute can be serial diluted by transferring to dilute one to the most common series doublet the dilution factor, which is transfer. This dilution can be done in micro filter plate and test tubes depending on the volume. हमने डाई को लिया, उसका जो 1 ml जो है हमने डाई का लिया और 9 ml लिया वाटर का, जिससे कंटिन्यूस हम सीरीज बाई इसको एक्सपेरिमेंट को करते गए और हमें लास्ट में एक 10⁻⁻⁶ जो है वो डाइल्यूशन हमारा तैयार हुआ. इसकी बात करें स्प्रेडिंग मेथड की, क्या स्प्रेडिंग मेथड क्या होता है, हमने ये बैक्टीरियल सॉल्यूशन लिया, उसको जो है हमने स्प्रेड कर दिया. उसके बाद में हमें डिफरेंट जो है कॉलोनीज मिली माइक्रोब्स की, जैसे आप देख रहे हैं डायग्राम में. फिर उसके बाद बात करें हम सेकंड टेक्निक की बात करें स्ट्रिकिंग मेथड. स्ट्रिकिंग मेथड में क्या करते हैं, उस कॉलोनीज में से कुछ कॉलोनीज को पिक किया जाता है और इनोकुलेशन के थ्रू हम क्या करते हैं उसे स्ट्रिक करते हैं. जिसे इस डिफरेंट मेथड दे रखा है, ये मैंने स्ट्रिक मेथड किया था यहाँ पे.
एशिया
और
को
यहाँ एक बैक्टीरिया का जो है एक स्ट्रिक दिखा मैं स्ट्रिक मेथड. फिर हमने माइक्रोब को आइसोलेट किया. ये ग्राफ है माइक्रोब का जहां हम बैच स्टडी करते हैं. तो हमने देखा कि कुछ स्पीशीज है माइक्रोब की जो हमें डाई के रिमूवल के बारे में ज्यादा जो है एफिशिएंट रिजल्ट दे रही थी। जैसे स्पीशीज 2 की बात करें, उसने लगभग 93 के अराउंड जो है हमें रिमूवल दिया। डाई को रिमूव कर देती है, डिग्रेड कर देती है। तो इस तरह से हमने माइक्रोब को आइसोलेट किया सबसे पहले। फिर हमने क्या किया, पायरोलाइसिस किया। पायरोलाइसिस क्या होता है, ऑक्सीजन की एब्सेंस में किसी भी प्लांट या किसी भी बॉडी को हम क्या करते हैं, बर्न करते हैं और नाइट्रोजन इसमें फ्लो किया जाता है। ऑप्टिमम कंडीशन की बात करें एक्सपेरिमेंट में, तो हमने पीएच 7 to 9 लिया है, टेम्परेचर 3 to 37 लिया है, और वर्किंग आवर की बात करें तो 3 days हमने लिया है। तो रिमूवल जो हुआ हमारा डाई का 60 to 85% रिमूवल हुआ है। ये आइकोनिया प्लांट है, जैसा कि हम इंडिया के
Rakesh,
हर
you have
वाटर।
1 minute left. Please complete with 1 minute.
Okay, sir. So this is Iconia plant, and biochar is prepared and then immobilize and biodegrade. These are a bioreactor. We ready the bioreactor and for dye removal. Then this is a data. Then we take the biochar, and initial dye concentration is 5 ppm. Then this is a literature and review. After that, ऑब्जेक्टिव की बात करें हम, तो माइक्रोबियल असिस्ट डाई जो है उसका कलर रिमूवल करना था हमारा ऑब्जेक्टिव। दिस इज करेक्ट, दिस इज अ फाइनल रिजल्ट ऑफ अवर डाटा, and this is a removal. This is a result of my data. The 63% dye are removed, and outlet concentration is, initial concentration is 5 and outlet is a 185 around concentration. Conclusion की बात करें, Iconia biochar bead is a packed bed bioreactor, offer a promising low cost solution for reactive orange biodegradation, outperforming conventional method via adsorption enhancement microbial action. These are reference. So this is my presentation according to my research work.
So am I
Hello?
audible?
Yes, sir.
Thank you, Rakesh ji, for your presentation.
Okay, thank you so much. Can I leave the meeting, sir?
I have a couple of queries.
Hello?
Hello.
The volume has echo, so sound is repeated.
No, but you have to come to the mic.
Hello?
We just have some questions for you.
Yes, ma'am.
So, what was the role of bacteria?
Sir, bacteria is a degrade the dye. The bacteria use a dye as a food source and then degrade in a simple compounds.
So, have you checked the compound using the GCMS or other technique?
Yes, sir. GCRMS techniques with some simplest compound CO2 like CO2 compounds, then some inorganic compounds are degrade because dye is an organic compound. Bacteria degrade this dye.
But CO2 is a gas; it's not a liquid part.
It is a not a compound, but is some sir different compounds we analysis through this process.
Okay, okay, thank you.
Okay, thank you. You may now leave the meeting if you please.
Okay, thank you so much, ma'am. Can I leave the meeting, ma'am?
Yes, you can leave the meeting.
Next up, we have Ashika Bhanu. If you can please start with your presentation. Ashika, you're not audible. I think you have not joined the audio.
Yeah, now it is audible now.
Yeah, you're audible now. You may start. Your time starts now.
So, good morning, everyone. Myself, Ashika Banu. I'm in second semester of my MTech Biotechnology from B.S. Abdur Rahman Crescent Institute of Science and Technology, Tamil Nadu. So my research title is Development of Chromophore for the Rapid Detection of NDM1, which is New Delhi metallo-beta lactamase 1 producing microbes. So NDM1 is New Delhi metallo-beta lactamase is referred as NDM1, and it was first reported in the Swedish patient who has taking treatment in New Delhi. So the name came as New Delhi beta metallolactamase 1. So this carbamase is a hydrolyzed antibiotic. Basically, for antibiotic resistance came because of this. This carbapinins are antibiotic given to the patients who are suffering, who are very much suffering. So that antibiotic also this enzyme will cleave. So in the active site of this enzyme, there will be two zinc components that will be helping in the cleaving of the antibiotic, and the gene encodes the NDM1 is BLA NDM1 gene. So it is basically majorly found in two bacteria, Escherichia coli species and Klebsiella pneumonia species, and there are over 70 variants are there. So mechanism of the NDM1 is that this is the antibiotic. So in the active site of the NDM, if you see, there are two zinc molecules that will cleave the antibiotic and which will make it inactivate, not work. So there are four major objectives of this research. The first is to develop a chromophore-based probe in such a way that it should selectively bind to the NDM1 and it should not bind with other enzymes like VIM1 and IMP2 because the NDM1 will contain L3 loop, which is very flexible, whereas the other two enzymes will not contain that. So if we design a larger probe, means it will directly target the NDM1 alone. Second one objective is to check the binding affinity using molecular docking, and third is to synthesize the probe which has shown good result and do in vitro studies. Then the bedside diagnostic tool was ready. So methodology is that first we have to select the 3D structure of the NDM and design the chromophore against the active site of the NDM and molecular docking studies. Then the synthesis of the chromophore and its in vitro performance, and then the cost-effective bedside diagnostic tool. So these are the 6 different probes that have been designed, and for that the protein is selected. The protein ID is 6XBF and the coordinates and the dimensions have been fixed. For this, the docking studies have been conducted for the 6 different probes. Among the 6 different probes, the probe number 5 has shown good binding energy, that is minus 10.82 kilocalories per mole. So this is the binding energy of that. And this is the active site amino acids present in the active sites. So mechanism of the probe is that it should, if the NDM1 is present in the patient, means it will cleave the probes and into 4 different sections. That is, this one is antibiotic region, this is a linker region, and this is a fluorophore region, and the CO2 will be liberated as byproduct. So if the probe is intact, means it will not glow. So there are, we have to synthesize 3 different portions, that is fluorescent region, linker region, and antibiotic region. As a whole, we can get a chromophore probe. There are 3 derivatives for the fluorescent probes. The first one is 4-methyl omliferone. The starting material was reacted to form this, and we can see the product has formed using TLC. Again, in the same way, we have synthesized 7-hydroxy, 3-carboxy coumarin ring, and 3-nitro 7-hydroxy coumarin rings. So for the linker, we have used 4-nitrobenzaldehyde, and that was reduced to form the 4-nitrobenzyl alcohol. Then for antibiotic, we have using imipenem and meropenem rings. So in future, I'll be joining all that three component and doing the in vitro studies. And my main target is to design a diagnostic bedside tool for a selective detection of NDM. Now it is available. Like if we take test, it will take around 1 or 2 days to get the result. Meanwhile, the patient will transmit the NDM1 to other patients through this is a bacterial contamination. So if we do rapid detection, means we'll separate the patient from not spreading to other ones. So thank you.
Thank you, Akshika. Am I audible?
Yes, sir.
Can you please explain the role of prosthetic group which is present around the Jing Center? Yeah.
So the zinc, there are 2 zinc active molecules that will cleave the ring present in the antibiotic.
So what is the role of the imidazole unit which is attached to the Jing Center?
So I'll have to look into it, sir.
Okay, thank you.
Thank you, Ashika. Is Satyam Kumar present in the meeting?
Is Satyam Kumar present in the meeting? Satyam Kumar? Okay, we'll go to the next candidate. Is Sonia Bhankor active in the meeting? Okay. Then we will go to the next candidate. Is Renu here? Renu?
Yes, sir. Am I audible?
Renu Yadav, you're not that audible. Please try speaking to your mic again.
Am I audible, sir?
Yes, you are audible.
You may start the presentation now.
Is my screen visible to you?
Yes, it is.
Okay.
You may start now.
Sure. Just a second.
Renu, you may start your presentation.
Just a second, sir. Sure. Hello, everyone. Dr. Ray. I'm here to discuss my work, that is cyclodextrin-based nanocomposites for the removal of organic dyes from wastewater. So here we have synthesized a photocatalyst, which is generally a nanocomposite, and which is mainly we are applying that photocatalyst on the wastewater to remove the impurities. So this photocatalyst which we are preparing, the size becomes in the range of nanometers. So this is a nanomaterial, and further we have converted it into a nanocomposite by combining it with the material that is cyclodextrin, that is oligosaccharide. So this procedure follows heterogeneous photocatalysis, means the removal of the pollutants from the wastewater. After the removal, the catalysis will we can easily separate the catalyst. And the process we are following is photocatalysis. So the initial oligosaccharide cyclodextrin that have the property that have separate properties, that this is water soluble, tasteless, colorless, odorless, non-toxic, biodegradable, biocompatible, and non-carcinogenic. We that have a very good significance or the property that it have yes, this is structure. It has two types of nature, that is hydrophobic as well as hydrophilic nature. Hydrophobic nature, which is generally we can say that water repelling nature. So and hydrophilic, water loving. So it can work on the both type of impurities, like polar impurities as well as non-polar impurities. As in case of water, we have different type of properties like biological, inorganic, and organic. Organic impurities, generally, if we discuss about the dyes that have most of the non-polar nature, and in case of heavy metals, they are polar in nature. So we are working on with the aim that there should be a photocatalyst which can work on all types of impurities at the same time. So this is our base oligosaccharide. This base is modified with some metal oxides and some capping agents. In this article, in this work, we have synthesized initially a hybrid metal oxide, zinc oxide, iron oxide, and after that we have combined the hybrid metal oxide with beta cyclodextrin, and initially beta cyclodextrin was combined with EDTA. Functionalization was done with EDTA, and then the this nanomaterial was synthesized by following the by using this procedure. Initially, iron oxide, zinc oxide, hybrid metal oxide was synthesized by using the precursor ferric nitrate, and for zinc it was zinc nitrate, and then we have prepared separately EDTA beta cyclodextrin, and then we have combined the both. So this is the experimental procedure we have followed. This procedure follows generally stirring, simple maintaining the pH, and simple processes removing the impurities by calcination. Then the prepared nanocomposite was characterized by using different analysis techniques. Just for example, PXRD. PXRD, we have analyzed the sample with via PXRD, and it was observed that the size that was calculated for the hybrid metal oxide Fe2O3 zinc oxide was 262.5 nanometer, and it was for EDTA beta cyclodextrin Fe2O3 zinc nanocomposite, it was 271.6 nanometer, and there was an enhancement in the peak which favors the formation of the nanocomposite after the addition of EDTA beta cyclodextrin. Then PL photoluminescence spectroscopy was also carried out, which is generally gives us the idea about the recombination of the electron-hole pair. As we know, in the case of photocatalysis, when the photocatalyst comes in the contact with the pollutant, its main role is to decompose the pollutant by creating the electrons and the holes. And as the speed of the production of electron and holes increases, the degradation also increases. So from PL, we can see as the from PL, as higher the peak, it means there is a higher recombination rate. So if we have a higher peak, it means recombination rate is high. So we for photocatalysis, we have to restrict that rate. So it means that after the addition of the EDTA beta cyclodextrin, the recombination rate has been decreased. It means restriction that what we want, that is working as this. Now, after that, band gap was also calculated, and it was found that after the addition of the beta cyclodextrin EDTA material to the hybrid metal oxide, band gap was reduced to 1.6 electron volt, and which is a great major factor for the photocatalysis. Then photocatalytic activity was carried out by using the material on initially a dye which is malachite green, and we have the absorption curve. It was observed that it was the experimentally the material was kept in the contact with the dye for 3 hours. Time to time readings were taken, and then we have the depressant degradation curve, and it was observed that degradation was enhanced in case of after the addition of EDTA beta cyclodextrin complex. Then we have kinetics curve also, and it follows the first order kinetics on the basis of R square values, which is generally like a more accurate or more precise value that is near about that. In final, we have analyzed that for methylene blue, the degradation was 80% up to in 3 hours by the addition of this beta cyclodextrin EDTA, and it was enhanced by the as compared to the hybrid metal oxide. It follows the first order rate kinetics, and the R square value was more accurate than 0.98. These are the references. Thank you.
Thank you, Dr. Renu, for presentation.
Thank you, sir.
So, have you identified the product of degradation?
Yes, sir. So we have worked on the product, like we have signed the samples for the BOD and COD, so that from that data we can analyze how about our products, that initial how much dye is left, or we are just left with the materials. Because see, if that is converting to H2O and CO2, then in the final amount which we have, or final solution which we have, we should not be left with the dye. That dye concentration should be reduced, and we can see the change color also, change in color also with the time.
So, how do you make sure that CO2 is not coming from CD and it's coming from the dye?
Yes, sir. So that's why, as I said, we know that cyclodextrin has two types of function group, that is primary and the secondary function group, and that is a very stable, and this compound has a solubility in water, but that is not too much soluble. A limited amount of solubility is there, and for that part, we have initially done the capping with that, capping and the capping with the citric acid, and that functionalization with the EDTA. So we are not and that have a decomposition temperature is above, which is we are working on that condition. So that is above all these conditions. So we have the positive factor about this, this will not degrade in that limit.
Okay, Dr. Nenu, thank you.
Okay, thank you, sir.
So for the next presentation, can we have Satyam? Are you here? Next, Subhash Kumar Jha. Gaurav Singh.
Yes, ma'am, I'm here.
So now you may present. You can start your presentation. Gaurav, please start the presentation. You're not audible.
1 minute. Now I'm audible?
Yeah.
Okay. My screen is visible, no?
Yes, yes.
So good morning, everyone. Myself, Gaurav Singh, registered here in MIT University as research scholar under supervision of Dr. Kamal Nayan Sharma. I'm going to present my work on synthesis and cytotoxicity evaluation of half-sandwich ruthenium complexes of pyrazole-based chalcodino-ethyl ligands. As we all know, ruthenium complexes are widely studied for catalysis and biomedical applications. Going through the literature review, it was found that Rogala et al. Synthesized ruthenium complexes with benzimidazole ligands. They showed anti-bacterial activity, and more important part is that they are non-toxic to human cells. And similarly, Garja Garja et al. Designed safe-based ruthenium complexes with tunable anti-cancer activity. And Similarly, Singh et al., they have not prepared ruthenium complexes, but they have reported nickel complexes of safe bases. And also they are showing promising antibacterial and antifungal activity. And N-heterocyclic and N-heterocyclic chalcogen-based ligands were reported earlier. But those were more studied only for catalytic activity, and therapeutic and detection activity are yet to be studied. So there was a research gap. This motivated my present work. Objectives of my work are to synthesize an heterocyclic organochalcogen ligand, characterize ligands by NMR and other techniques, and prepare their ruthenium metal complexes, and if possible, other metal complexes also, and evaluate biological activity and detection and therapeutic applications. So, going for the synthetic part, I have prepared 5 ligands: L1, L2, L3, L4, and L5. So L1, L2, and L3 are more or less similar. Only part is different chalcogen atom is there. The starting material used was common for them, 4-bromopyrazole. And first step was N-alkylation, and then uh, reduction of this sulfur, selenium, or tellurium powder. And After nucleophilic substitution or ligand and purification also, our ligand was prepared. L1, L2, and L3 was prepared on the similar manner. And similarly, L4 and L5 were also prepared more or less similar, but only difference is there. Benzimidazole moiety is there. So starting material was used benzimidazole, and alkylation part was there similar to the prior L1, L2, and L3. And this chloroamide moiety was also incorporated to the system, so that this N-heterocyclic carbene moiety type of ligand is prepared. So moving towards the characterization part, I am showing L2 and L3 characterizations by proton NMR and carbon NMR. So clearly there are two triplets that is for these two two methylene carbons, and here is for 4 carbon for aromatic system, 4 hydrogen for aromatic system. Similarly, carbon NMR was also done, and we can clearly see the pattern of the peaks, which we can clearly identify the structure. Similarly, it was for L2, and L3 was also similarly done. L4 and L5 currently not characterized, so it is under progress. I have prepared complexes with L1, L2, and L3 ligands, and crystal structure is solved, but currently I cannot provide the single crystal XRT data. So basically, from the single crystal XRT, or this half sandwich type of complex is formed. So, my future plan is characterization of other synthesized ligands, and synthesis of some other metal complexes, characterization of other metal complexes, and in vitro biological study of the prepared complexes for detection and therapeutic applications. Thank you. That's all from my side.
Gaurav, am I audible?
Yes, sir.
Okay, so Gaurav, you talked about a carbene ligand, right?
Yes, yes.
So there are two kind of possibility in case of carbene, singlet and triplet.
Yes, sir.
So what was the nature of your carbene? Is it a Fischer type of carbene or Stock type of carbene?
I have to go into this, sir. I have just prepared. I have not much studied on this.
Okay, so what kind of methodology you will use to identify the nature of the carbene?
UV visible spectra for singlet and triplet transitions.
Okay, okay, Gaurav, thank you.
Thank you, sir.
Thank you. Now you may leave the meeting. Next candidate, Satyam.
Yes, ma'am.
Share your
Present
screen,
my
please.
screen. Yes, ma'am. Hope it's visible, ma'am. So, good
Not visible.
morning.
The camera is visible.
University. Is it visible?
No. No, no, no.
Hello, am I audible, ma'am?
You're audible, but you're not, your screen is not sharing.
My screen is not.
I think you have a network issue. Please try to connect later on. So for the next presentation, we can call Subhash Kumar.
Yes, madam, I'm here.
Okay, share your screen.
Yeah, please. Oh, okay. Is it my screen is visible, madam?
Yes,
Okay.
you can
So,
start.
good morning, everyone. Myself, Subhash Shah. I'm a PhD scholar, and my supervisor name is Professor Dr. Seema R. Pathak. Well, my topic is next generation molecular tailoring of the tucatinib to overcome its therapeutic resistance. So, what are the therapeutic resistance? When we are considering about the therapeutic resistance, it is kind of a mutations happened into the body when we are taking to the drug. At the same kind of, if we see the tucatinib is a HER2 directed tyrosine kinase inhibitor, where the HER2 positive metastasis breast cancer, including patients with brain metastasis. Along with the tucatinib, if we take one more molecule that is a sorafenib, this is also a kinase inhibitor drug approved for advanced renal cancer, hepatocellular cancer, and uh, thyroid carcinoma. So, however, acquiring resistance of kinase domain mutation, activation of compensatory pathway, and tumor heterogeneity limited during the therapeutic resistance. So, we have modified this both tucatinib and the sorafenib moiety. And when we look into the moiety, we find the rigid compound side, a side chain of the rigid compound of this molecule. We took that one part, and from the sorafenib also we took a 4-hydroxy aniline part of that moiety, and we modified it into our moiety. So which reduce the resistance. So when we synthesize according to the structure, we synthesize almost 25 plus moieties and characterize it by the NMR mass. And when we see it, the impacts on the docking, uh, on the molecular docking side, we find the sorafenib and our synthesis moieties working similar, similar to the protein molecules. That is, one UWH protein molecules works even when we dock this moiety compared to sorafenib, the level that the score of our moiety synthesized moiety NCE1, it is more potent than sorafenib. So our next target is we will take this our moiety, and we will uh, go through the protein uh, enzymatic evolution also, like this one. So here you should find the synthesis moiety which have the some particular linkage like pyridine triazole ring. You can find here the pyridine triazole ring, which uh, which on activity it will find that during the assimilation, during the drug assimilation in the body, it will not metathesis at uneven places. Like it will not degrade at before its targeted position or targeted site. So this is the advantage of our synthesized NCA moiety. And you can also say that this will give the moiety to be enhanced for the further generation. So like this, we prepared the moiety. Here I can not share you the perfect scores of this docking data because this has not published yet. So on future perspective is the systematic molecular refinement of 2-continue, giving a medicinal chemistry and translational pharmacological response for the rational approaches to overcome therapeutic resistance. So here you can on images also you can find that the NC1 interaction with 1UH residues. So how it is interacting with the protein. So that's it from my side.
Thank you, Subhas, for the presentation. Can you
Yes, sir.
explain the design criteria of inhibitor?
Design criteria of inhibitor.
Right.
Okay, so when once we choosing the compound like structural based drug design, when you're taking that structural based drug design and we are injecting our moiety structure and we finding the in the structural based drug design we are finding the computational structure of the same protein and finding where the active site is there, right? And how our moiety at the computational base, how our moiety will be bind to that the active site and how long it will be remain, that we are finding from this computer based drug design. And by this way we are finding the inhibition, whether inhibition is there or not, so that we are comparing with the moiety whatever the present in the market.
Okay. Thank you, Subhasji. You can.
Thank you, thank you.
Thank you for the next presentation. Can we have Dhanya Govindan?
Ma'am, am I audible? I have connected with another network, so can I show my?
Yes, you can share.
So, good morning, all. My name is Satyam Kumar, and I am from Bikram University, Ujjain. And this study is about impact on ESG and sustainability disclosure on wastewater treatment approaches by the industry.
There's lot of background music noise.
Uh, now is it fine? No, it's coming from another source because I can also hear it.
Okay, okay. Other participants, kindly.
Yes, now it's clear, ma'am.
Yes, you can start.
Yes, ma'am. So, according to the Central Pollution Control Board, a significant portion of industrial wastewater in India are not being treated. Either it's discharges in the any river or any river or land, which is remain untreated, and this is a significant environmental issue in India. So, contributing to the pollution and ecological degradation related to this wastewater. According to the national estimates, India generate over around 72 million liters wastewater per day, and only 28% of wastewater is being treated currently. So so around 80 to 72% of water is not being treated on daily basis. So a study suggests the industries to adopt advanced wastewater treatment technology, which can be improve the efficiency of around more than 50% uh, efficiency and reduce environmental impact by 50% as well. So Our study investigates whether these disclosures are merely reported mechanism or actually drive improvement to the wastewater practices. So this is the research problem. While ESG reporting has increased significantly after the introduction of mandatory BRSR guideline regulated from SEBI, there is a still empirical evidence linking to this actual technical improvement, and the central question is remain same: do ESG disclosure lead to real improvement in wastewater treatment? So we have find gap that limited research connecting these two. One is ESG disclosure and second one is wastewater treatment technology. So while ESG reporting is increased, there is limited research which is related to these two linkage of these two aspects. So for that, actually, our study has examined three key objective. First is uh, how ESG disclosure impact wastewater treatment system, and second is compared to the impact across all the different industrial sectors, and third one is the excess technological portion pattern, which is advanced technology. So this is the methodology which we adopted. We adopted mixed methodological approach, which combining qualitative and quantitative analysis, and we focused. So our approach is to access data from BRSR report, IR report, and CPCB records from industrial website. And we have considered sectors like textile, pharma, chemical, mining, and energy. So there are some key findings, key technology which can be used in the industries like that. So we analyzed to adopt the key wastewater technology, including zero waste to land, zero liquid discharge, membrane bioreactor, reverse osmosis, and closed loop water reuse system. And this technology present the industry shift toward more sustainable practices. For example, if we talk about zero liquid discharge, so this is the mandated this is being mandated now. It's not completely mandated, but this is being mandated for sectors like textile, pharma, and all.
Please don't exceed 5 minutes. You may
Okay, thank
conclude.
you. Yes, ma'am. So our finding shows the clear difference where pharma sector demonstrates the highest compliance, which align a strict regulations. Textile sector a strong implementation on ZLD system, and while the chemical and mining improves their performance, because on the other hand, mining and energy sectors are still evolving with gradual adoption in these days. So these are the key findings. First is clear positive association between ESG quality and ESG disclosure quality, as well as the wastewater treatment system. So firms with more detailed and transparent reporting, currently we can see they are more investing on the capex or advanced treatment system. So this suggests that ESG disclosure as an indirect regulatory mechanism influence corporate behavior as well. So these are some discussions. This aligned with the growing influence of ESG focus investors also. Transparency, it increases transparency and accountability too. These are these combining s- um, uh, these two sectors combinedly, uh, it will give best result. And finally, to conclude this, ESG disclosure are a powerful drivers in nowadays in wastewater treatment system, because of the disclosure, they are investing in the capex, and they are improving their wastewater quality as well to show to the investor and customers. And this is not sufficient because there are lots of regulatory requirement as well. They are also supporting to disclose more transparently and efficiently. And third is it improves the sustainable development goal also it supports. Thank you.
Thank
Do you
you,
have any
Satyamji,
question?
for the presentation. So I am wondering what initiative would you take for the improving of the compliances?
So sir, there are lots of things we can do. There are because of the investor pressure and the customer pressure. There are lots of policies which are coming nowadays, like URD. If we are exporting anything to outside within the European Union boundary, and because of BRSR, and also there are lots of pressure from investors. So we need to be transparent in terms of reporting anything related to water and wastewater treatment system, what trends we are following. And there are lots of agencies also, or lots of framework which we are adopting, like GRI, SASB and all. They also want to disclose the trends. Yes.
Okay, thank you, Satyamji. You can leave.
Everyone, please try to finish your presentation within 5 minutes. It not may exceed. So for the next presentation, can we have Dhanya Govindan?
Yes, ma'am.
Okay, start your presentation.
Ma'am, is my screen visible?
Yes.
Okay. Good morning, everyone. I'm Danya, studying in BS Abdurrahman Crescent University. So today I'm presenting my work on phytochemical characterization and of alkanet tinctoria and formulation of a herbal lip balm with comparative phytochemical evaluation. So this study focuses on developing a natural alternative to synthetic lip balms while ensuring comparable performance. So coming to the introduction, cosmetic products have been used since ancient times, but recently there is a clear shift towards natural formulation due to safety concerns. So lip balms are one of the most widely used common cosmetic products. It is not only used for the appearance but also for protection and hydration of the lips. So, however, most commercial lip balms contain synthetic colorants, additives, and petroleum-based ingredients. So, these may cause irritation and long-term concerns. So, this study, I focused on developing a chemical-free herbal lip balm using Alkanet tinctoria, which is known for its natural red pigment and therapeutic properties like antioxidant, anti-inflammatory activities. So, this is the plant. So Alkanet tinctoria belongs to family Boraginaceae. Its root produces a red to violet dye. So the color is due to the presence of compounds like alkanin and chiconin. So these compounds are biologically active, which is known for the antioxidant activity as well as anti-inflammatory effect, and it also has antimicrobial properties. So because of these properties, it is suitable not only as a colorant but also as a functional ingredient in cosmetic formulation. So, to the problem statement, as I have already mentioned, the major issue with the existing lip balms is their dependence on synthetic ingredients. So, for that, the herbal lip balm exists. They often lack stable natural pigment, proper scientific evaluation, and comparable performance with commercial lip balms and all. So my aim is to formulate and evaluate a herbal lip balm using this, and my objectives are I performed extraction and phytochemical analysis, and then I have characterized the compounds using GCMS, and then I formulated the lip balm and evaluated its physical chemical properties, and then I compared the formulated lip balm with the commercially available lip balms. So coming to the methodology, the first plant material was collected and dried and then powdered. The extraction was carried out using two techniques. One is aqueous extraction. Another one is acetone solvent one. Secondly, the phytochemical screening was performed, followed by GCMS analysis to identify the bioactive compounds. Third, the lip balm formulation using a natural basis along with the plant extraction. Finally, the product was evaluated for parameters like organoleptic properties, color, odor, texture, melting point, spreadability, and then softening point, and then safety and stability, and then pH determination. So this is my qualitative analysis result. So this confirmed the presence of alkaloids and sterols, which are known to contribute to biological activity. Sterols in particular play a major role in improving skin barrier functions and hydration, so making them relevant for topical applications like lip balm. The absence of tannins and saponins is actually beneficial, as these components are sometimes can cause dryness and irritation to sensitive skin like lip skin. So overall, the phytochemical profile supports the suitability of the extraction for safe cosmetic use. So coming to the GCMS analysis, it identified several major bioactive compounds in the alternate interior, including beta cytosterol, camposterol, 1-hexadecanol, and 1-aptadecanol. So beta cytosterol and camposterol are plant sterols, which is known for anti-inflammatory and skin conditioning properties. Hexadecanol and octadecanol or long-chain fatty alcohol, which acts as emollients and help improve the texture, moisturize retention, and stability of the formulation. So these compounds are not just present; they directly contribute to the both the functional and physicochemical properties of the lip balm. So these are all the F1 and F2 lip balms which I have formulated. F1 is this white color, which is in which I have not added the alkanoid. So these are all the comparative evaluation I have made using the F1 and F2.
You may
So
conclude your presentation now.
yes, ma'am, I can conclude. So these are all the comparative results I have made with commercial and the formulated lip balms. So which showed the overall, the result demonstrated that the formulated lip balm has successfully integrated natural pigment and bioactive compounds from alkanet tinctoria. And although slightly lower the temperature stability to commercial product, it improved a safer and more natural alternative without compromising the functionalities. So the future scope is to optimize texture and stability, long-term shelf life studies, and scale up in industries and additional plant-based activities inclusion. So that's it, ma'am. Thank you.
Thank you, Dhanya, for presentation. So
Thank
have
you,
you
sir.
quantified all the component present in the lip balm?
Thank you. Components in lip balm.
Like the carbohydrates and other things. Have you quantified?
No, sir. I did qualitative and GCMS only. So GCMS have, identified more components which is present in the plant, but qualitative I have did
So
for
if you have larger content of alkaloids,
yes,
then it may cause
alkaloids.
then it may cause skin irritation, right?
Yes, sir, but there is no chemical usage in the formulation as it is only natural ingredients, so that will not cause that much irritation or irritation like things. So I have done a preliminary uh, human test by just by applying in people's lips. So they have said there is no irritation and like that.
Okay, Dhanya, thank you.
Thank you, sir.
Everyone, please take care of the time limit. It may not exceed more than 5 minutes. So for the next presentation, can we have Pradeep Agrawal?
Yeah,
Okay.
my name is Pradeep Agarwal. Am I audible?
So your screen is not visible.
1 second, I will share the screen. Is it visible now?
Yes, sir.
Okay. So I am Pradeep Agarwal. We manufacture high density polyurethane foams for footwear application. And very recently, say about 2 years back, I have joined for PhD in my alma mater, Delhi College of Engineering. I did my graduation in 1981, and I thought of doing PhD in the subjects in which we are already doing commercial productions. So that is basically high density flexible polyurethane foam for footwear applications. Now, flexible foams, they are everywhere. You can find them in insoles, in mattress, in any comfort application, you can find flexible foams. So my guides are Sagar Maji. In fact, he was my batchmate in 1981, and Professor Roli Parwar in chemical engineering, the DTU. Now, my why high density foam? We targeted density of 100 kg per 110 kg per meter cube. We sell maximum insoles and many global brands, they are using this kind of densities. Now, most of the, in fact, if you see most of the studies, they had been done on low density foams and very high CNC loading, cellulose, nanocellulose loading 5 to 40%. And most of them, they were not based on industrial system. What we did was, basically, it was for a completely industrial system that we did. And what we did was we used two grades of nanocellulose, which were commercially available and of two different sizes, 88 nanometer and 95 nanometer, with different percentage of weight in the solution. And we tried from 0.25% to 2% in polyol. So that is ultra low loading we tried, and two different kind of sizes we tried in the existing polyurethane foam polyol. Now, the previous the previous was studies was basically less than 60 kg per meter cube. That was not good for the comfort applications. Normally, they were for this insulation application. Or what some for high density, some rigid PU foams, they had done some work, but that was very, very high CNC. They had used 5 to 40%. And their route was taken as reactive material. They substituted polyol with CNC or isocyanate with CSC, and they use it. So our aim was to use it in the application of footwear. So that's what we did. What we did was we took the recipe which we had been already been using, and in that, like polyol, surfactant, catalyst, etc., we are already mixing to them, and that we added 0.25% to 2% of different kind of these two different kind of nanocellulase, and then in a closed mold we poured it, and then we made out the foam, and then we removed the skin and then sliced it into different thicknesses to test the materials in tensile strength, tear strength, elongation, rebound. And then we did FESEM. And from FESEM, we used ImageJ software to calculate cell count, pore size, circularity, solidity, porosity, and these kind of things. TGA we had done. So these tests we did. And what we found out was that quite interesting. In fact, what we did was in type A, the tensile strength we found in type A, that was in the lower size of nanocellulose, it increased to quite an extent, 41%. And in B, where the size was a bit bigger, we found the improvement was 28 to 30%. And there was a plateau effect after say about 1% in polyol. And similarly, we found in tensile tear strength also, there was an increase. 38% was the increase in the type A. So we found that mechanical strength. Okay, I will try to finish it as early as possible. And then in FESM, what we found was the cell counts and pore area, etc. They were not cell count increased a lot, and pore area they decreased. So that showed basically and solidity, etc. That was maintained, and porosity also increased. So porosity is also an important part in the insoles manufacturers. Now we did TGA also, and in TGA we found not much of the changes. A small improvement was there, which we found, but it could be because of the experimental variations and like that. So ultimate these results was type tensile strength. It increased to 41%, tear strength increased to 38%, porosity increased, and the difference between the particle size, there was a difference of 7 nanometers in particle size, and that made a significant performance divergence. And the rebound resilience and compression set, which were also important part in footwear insole, they were maintained. Thermal stabilization was marginal. So these were the results we obtained. Thank you so much. That is my presentation.
Thank you
Any
for the
questions?
fast presentation. What could be the possible explanation of the increased porosity?
The increased porosity was because of the morphological changes. Could be because the cell sizes became smaller, and so the gaps, it has an open cell structure, so gap increased. The gap increased, and that could result into porosity. So that is the evaluation of that.
Okay, sir. Thank you.
Thank you.
Thank you for your presentation. We'd like to call Akansha Soni to present next. You have 5 minutes for your presentation. We'll let you know at the 1 minute mark so that you can do your conclusion.
Is my screen is visible?
Your screen is currently visible, but it's showing a white page.
My screen is visible or not?
No, it's not.
Okay. Let me check, sir. 1 minute, please.
This is visible here.
Okay, 1 second, please. Is it visible?
Yes, it's visible now. You
Okay.
may start your presentation.
Okay, sir. In a world where environmental concerns are growing, the way we approach the chemical reactions matter the most. A very warm good morning to everyone. My name is Akanksha Soni. I'm from MNIT Jaipur. Today, I would like to give an oral presentation on sustainable water-promoted iron catalyzed one-pot cascade hydroaminomethylation of alkenes with nitrobenzenes. This is the table of content. I will explain one by one. So amines are everywhere, from pharmaceuticals to agrochemicals. Nearly 70 to 80% of drug molecules contain amine functionalities. For that instance, amines are most important, significant bulk and fine chemical and chemical industries. There are a wide range of amines are key component in the production of pharmaceuticals, biological active compounds, and dyes. If I'm talking about the global amine market, it was 6.12 billion US dollars in 2022 and expect to be a revenue of CAGR, which means compound annual growth rate of 6.3% during the upcoming forecast period. This is some important drugs which contains secondary amines. This brings us to the hydroaminomethylation reaction, which means basically a one-pot reaction which in multiple steps. That means it simply converted alkenes into just amines in 3 steps. First of all, alkenes converted into hydroformylation to form aldehydes, then condensation with amines to form Schiff base, or we can say that's imine, and ultimately hydrogenation to form amines. Basically, a multi-component reaction in just a single step. This hydroaminomethylation is considered as a most economic and environmental benign, which contain minimizing waste and process steps. So basically, it's considered as a sustainable reaction for the formation of amines through alkenes. These are some literatures based on hydroaminomethylation reaction. If you look at all these literatures, you find that around 70 to 80% of hydroaminomethylation reaction are from rhodium catalyst. Apart from that, they use additionally CO gas and hydrogen gas for the formation of aldehydes and hydrogenation reactions. And lastly, if we are looking into this, they are using ligands for the linear selectivity. That means they form major and minor products. That means linear or branch products. But for the linear product selectivity, they use additionally ligands. And lastly, they use all of these are considered to be a harsh reaction conditions. After getting loopholes in all these literatures and inspired by MI lab work, that means we are already working with the combination of iron catalysis and water as a source of reduction. That means they are considered the combination of iron catalysis and water as a reaction medium are considered to be a most sustainable and economical pathway to design and synthesize molecules. After getting inspired by all these reactions and the loopholes of the literatures, I found that I tried this reaction, hydroaminomethylation reaction, with the use of iron pentacarbonyl and water as a source of hydrogen. That means a surrogate source of hydrogen and a solvent also. So in my reaction, basically is a multi-component reaction. That means basically 4 type of reaction in a single vessel. That means alkenes to aldehydes. Nitrobenzene reduces meanwhile on the anilines, and these are combined to form Schiff base, and then ultimately reduction of this Schiff base to amines. So basically, these all 4 multi-component reaction in just a multi-step, in a single step. And I didn't use any ligands for the linear selectivity and no harsh conditions for this hydroaminomethylation reaction. And I tried this reaction with the large substrate scope. And more importantly, this is a 4-step reaction. It's a one step further from the classical hydroaminomethylation reaction.
You have
For
1
getting
minute left.
best, sorry, sir.
You have 1 minute.
Okay, I just concluded my work that after getting best optimizing yield, I try this with different substrate scope, just with different nitro and different styrene substrate scopes, as you can see here. And vinyl ferrocene we are also synthesizing in our lab, so we also doing with the vinyl ferrocene compounds. I synthesize with different selenium compounds because selenium is a therapeutic potential against several diseases and like anti-cancer antioxidants. For the confirmation of the data, I did characterization from proton NMR and the carbon C13 NMR spectra. And for the confirmation of water that hydrogen is a source, we did D2 experiment and we got success. And we did very we did try to control experiments for the confirmation of the reaction pathway. And lastly, this is a plausible mechanism of my reaction. And I just conclude my work that all my reaction is free from ligands, external, free from toxic CO gas and hydrogen gas. It operates at low temperature. Water is a sustainable solvent, which can be a great important in the chemical industries and replace, most importantly, the expensive noble rhodium complex with the iron complex. Lastly, I would like to thank my supervisor, Dr. Rajkumar Joshi, MNIT Jaipur, for CSIR Delhi for the financial assistantship, and lastly, MIT University Gurgaon for making me here, for giving me this opportunity to present my work. Thank you.
Thank you, Akanksha, for your presentation. So what was the technically reducing agent in your reaction?
So basically, I use water in place of many acidic source, or basically a reduction source. That means I replace all the harsh reduction source with the water. That means water is the only source of reduction as well as a solvent. This is the key point, one of the highest key point in my reaction. And second thing is, I use my reaction is without using any ligand. So basically a multi-component four-step in just a single vessel with a lesser amount of substrates, which means an atomic reaction.
So have you isolated any intermediate to support the fact that water is a
Yes, sir. Yes, sir. I isolate the reaction with, as you can see here on the screen, that we isolate amines and aldehydes, which confirms that there is reactions through hydroformylation reaction, and it is also confirmed by the HRMS spectra.
Okay, thank you, Akansha.
You're welcome, sir.
Thank you for your presentation. We'd like to call the next presenter, Anand Salvi. Anand Salvi. Next, we'd like to call Rakesh Patel. Rakesh Patel. Okay, we'll continue on. Sukhpreet Kaur. Sukhpreet Kaur. Sukhpreet.
Next up, we have Dr. Gurjinder Singh.
Ma'am, Devendra Singh this side. If someone is not available, can I present?
Please go ahead and present.
Hello.
Bhavna Vidani, you'll be next. Please stay ready after Devendra Singh.
Okay, sir.
Thank you.
सर आप स्क्रीन शेयर कर सकते हो?
Sure. Yeah, so my screen is visible.
Yes, you may start your presentation.
Yeah, okay, thank you. So myself is Devendra Singh. So my topic was the tailoring dispersion and amine synergist for improvement, packaging, inks, curing, and transfer properties. So this is my research topic under the guidance of Dr. Anirban Das. And for this past ST 2016, so my aim of the research is, this current research is to study the effect of variation of type and concentration of dispersant on the degree of dispersion. This will result in the reducing of pigment loading. It will reduce the migration of colorant into the secondary food packaging. Thus, it will help in reducing the environment impact. And so what is the dispersing agent? Dispersing agents are the used to disperse and maintain the stable state of dispersion of the mineral and organic particles suspended in the liquids. Their use is great value in the mineral industry, paper, board coating, ink industries, and many more, spray paintings. So dispersing agents having the very wide industrial scope. And now come to this chemistry part. The mechanism of this dispersing agent is, this dispersant provide the anchor group for interaction with pigment or filler surface. A charge is generated on the surface of this pigment, and the more repulsive force is generated between these pigment molecules. Hence, this repulsive force keeps this two tiny particles keep away. So when these two particles having the same charges, they repel each other, and the charges effectively forms a barrier to the closer interaction between the particles. And I have carried some experiment also in laboratory. So this is the just a process. What is the process of this stabilizing of the system? First is the wetting. Wetting is the process where this pigment need to soak for a particular period in the vehicle or medium. After that, grinding, And this grinding, I have carried this grinding by the triple roll mill, and several other methods are also available for this grinding. But here in study, I have considered this triple roll mill. And after that, we have checked this behavior of this stabilization. So in this next slide, this is the method how we prepare this slurry or premix, and then we go for this grinding method. So this is the grinding device. And come to the next slide. So this is the impact of this dispersing agent. So if we will see the grinding, so grinding was drastically reduced when we use this dispersing agent. And this flow property will also improve. This flow of this slurry was 3 centimeter without dispersing agent, and after that it was 8 centimeter. Even this color strength was drastically increased. This is 87% to 112%. So almost 25% there was increment of this color strength. And this is the optical density. So optical density was also increased. And now come to this effect of grinding. So this is the practical picture of this experiment on this left side. This is with a dispersing agent, and on this right side you can see there is a tiny lines are there. So this is the particle size of this pigment. So without dispersion, so there was the particle size was sorry.
You have 1 minute left.
Yeah. So I'm on the conclusion page. So this conclusion is that, so this dispersing agents supported in the wetting of pigment, making pigment soft and helping in the grindings, and also making system stabilized by the preventing the agglomerate formulation. So this is all about my.
Thank you, Devendraji, for presentation.
Thank you, sir.
What was the nature of your dispersing agent?
Nature. So, sir, it was the anionic dispersant.
So what will happen if you are substituting anionic with a cationic dispersant?
So sir, anionic or cationic. So my study is underway. So because here I am also considering the difference between the anionic or cationic. So because sir, cationic sometime may hinder us some pigments, because here I have considered this 57:1. So this just 1 minute. I would like to show the structure of this pigment. So here this is the structure of the pigment which I have shown on earlier presentation. So sometime what happens if we are taking the cationic pigments, so there may be some interaction, because this is also some having the cationic charges. So this study yet to carried out, sir. So what is the impact while we are replacing this anionic with the cationic? It is yet to be concluded. So I'll whenever this presentation, or I'll visit the university, then I'll submit some test report.
Okay, thank you.
Sorry, my screen was not shared. So sorry. Just I have shown you. Just wait. Structure. Okay, here. Yeah. So now it is visible. So this is the structure of this pigment which I have shown you in presentation. So I have carried the several experiments. So here I have taken this pigment. So this is the pigment name, and I have used this dispersing agent here. So this is the some results. So experiment number 1, 2, 3. They are showing increasing pattern of this color range.
All right. Thank you very much.
Thank you, sir.
We'd like to call Bhavna Vedani.
Yes, sir. Is my screen visible?
Yes, you have 5 minutes starting now.
Okay. Good morning to all. Myself, Bhavna Vidhani. Just a second.
Yes. Some participants of chemistry are not here.
Is my screen visible?
Yes.
Actually, it's not visible to me.
It's visible to us.
I'll stop sharing and then I'll share it again, please. It's not visible to me, yeah. Yeah. Good morning, all. Myself, Hafna Vidhani, Research Scholar, Chandigarh University, and Associate Professor in the Department of Physics, University of Delhi. I am working under the supervision of Dr. Sanjeev Kumar, Department of Physics, Chandigarh University. The topic of my presentation, the oral presentation for today, is green biointerface engineered mesoporous anatase TiO2 for rapid ciprofloxacin removal and antimicrobial applications. Now, this slide introduces the problem. The antibiotic residues are now persistent contaminants in the water systems, and they also contribute to antimicrobial resistance. Among them, the ciprofloxacin is especially important because it is widely used, strongly sorbed, and degrades slowly under natural conditions. The conventional water waste treatment often cannot remove it completely, so there's a need for a treatment platform that is not only efficient, but it also has to be reusable and environmentally safer. Now, why ciprofloxacin matters and what is the conventional treatment? Is it insufficient? And there's a need for a better photocatalyst. So the question was not just how to synthesize TiO2, but how to make it structurally useful for real pollutant removal. The main research gap is that many green TiO2 studies stop at the synthesis level and basic characterization level. So they do not clearly explain how the extract chemistry affects the surface architecture and ultimately the photocatalytic performance. Our approach was different. Our approach was different. We used LCMS to understand the metabolite profile of the plant extract and then linked that chemistry to the nanostructure formation. The objective was to design mesoporous anatase TiO2 capable of rapid ciprofloxacin degradation with an additional antimicrobial benefit. We did not treat the plant extract as a black box. Here I have shown the synthesis route. First, the Ricinus communis leaf extract was prepared using and it is used as the natural chelating and capping medium. Then titanium tetra isopropoxide was added and the pH was adjusted to 8.5 using ammonia. The mixture was then hydrothermally treated at 160 degrees Celsius for about 17 hours. Finally, the product was recovered, washed, dried, and obtained as Ricinus communis TiO2 nanoparticles. The extract here is not only eco-friendly, but it also helps to control the hydrolysis, nucleation, and aggregation. This slide explains the chemical role of the extract. LCMS showed the presence of polyphenols, glycosides, saponin-like species, and lipid-like components. These metabolites, they can coordinate the titanium for moderate hydrolysis and stabilize the growing nuclei. So instead of acting only as a green solvent, the extract behaves like a growth-directing biointerface that suppresses the hard aggregation and helps to generate the stable nanoscale TiO2 domains. This is the central novelty of the work. We then confirmed the structure of the synthesized synthesized material. XRD images, they showed the phase pure tetragonal anatase TiO2 with no detectable rutile or bruchite impurity. The broadened peak in RCTiO2, the first this A graph, it is for RCTiO2, XRD for RCTiO2.
Ma'am, you have 1 minute left.
Indicated smaller, crystalline domains. Williamson, Hall, and modified Debye-Scherrer analysis also suggested coherent domain sizes of about around 6 to 7 nanometer, while HRTEM showed the quasi spherical particles of about 10 nanometers with clear anatase lattice fringes. So structurally, we obtained a pure phase pure nano crystalline anatase system. This slide shows it highlights why the material is functionally attractive. RCTiO2 showed a high BET surface area of about 120.9 square meter per gram, a pore volume of 0.336 cubic centimeter per gram, and a average pore diameter of about 7.83 nanometer, confirming the mesoporous architecture. The indirect band gap of about was 3.01 electron volt, and the point of zero charge was around 6.49. In simple terms, we can interpret that the material has high excess surface and favorable charge behavior near the working pH.
Ma'am, please conclude as your time is over.
This is the main performance slide. We worked under the UV irradiance. The RCTiO2 achieved about 95% of ceprofloxacin degradation within 60 minutes. The optimum condition was 50 milligrams per liter catalyst at pH 6 for 50 milliliter per ciprofloxacin. The degradation, it follows the pseudo first order kinetics, and the highest apparent rate constant was 0.0537 per minute at the optimum catalyst loading. The best performance came near the pH 6, which is important because it is close to the practical water conditions. This is the mechanism. I'll just leave because of the shortage of time. And this slide, it shows the material that it is not only active, but it is also durable and multifunctional. We actually, the multifunctionality, I'll just conclude this slide also. This multifunctionality is useful because the contaminated water often contains both the chemical pollutants and microbial risk also. So next is the conclusion slide. To conclude, we successfully developed a plant-mediated mesoporous anatase TiO2 platform using the Ricinus communis leaf extract. The LC-misguided biointerface engineering helped us to connect extract chemistry with nanostructure and performance. The final material, it combined the rapid ciprofloxacin degradation, good structural durability, and additional antimicrobial activity. In future, this work can be extended towards the real water validation, mineralization, and toxicity analysis, and development-focused stability studies. Thank you very much for your patience. This is all.
Yeah, so Bhavna, I have one questions.
Yeah.
So you use now this anatase grade in of TiO2. So this anatase grade was made up of sulfate process or either by chlorine process?
No, sir. Actually, we are actually supposed to produce that. I mean, out of the because actually we intend to produce this anatase phase because it is actually highly useful out of the other the three phases that we have of TiO2.
No. My question is that because generally this TiO2 produced by the two route, chemically there are the industrial base, there are the two process of manufacturing of this TiO2. One is the anatase and second is the rutile grade. So you use in your study this is anatase grade. And anatase grade is further produced by the means it having the coating of the two coated by this TiO2 particle coated by the two process. One is the sulfate process and second one is the chlorine process. So my question was that, so which grade you used in your studies? Either it was sulfate process or chlorine process?
Sir, actually, we used this is a green process that we are making this TiO2. I mean, I am not very sure about the chlorine phase or the sulfate phase that you're asking about.
Thank
We
you.
actually selected the green route. So in the process, we got anatase phase, which actually we intend to get.
Okay. Thank you.
Thank you for your presentation. We'd like to call the next candidate. Is Karthik there? Karthik? We'd like to call in Sukhpreet Kaur. Sukhpreet Kaur. We'd like to call Mohini Gupta. Mohini Gupta.
Hi, good morning. Am I audible?
Yes, you are. Sukhpreet, if you can hear us, please be ready for presenting next. Mohini, you may go ahead.
Okay, this is visible to you all.
Yes, it is.
Thank you. Good morning. My name is Mohini, and my oral presentation topic is combustion synthesis of manganese doped calcium fluoride nanocrystals, effect of dopant concentration on structure and morphological properties. Sorry. This is the outline of my presentation. I will go one by one within my presentation. Why this is going like this? So before going into the synthesis, I will introduce about the structure of the fluorides. Fluoride is a halide anion having a chemical formula of F minus. Fluoride ion is the simplest inorganic monatomic anion of fluorine with basic properties. Fluoride structure generally shows the structure as shown is in the figure. X tends to occupy the A tetrahedral interstitial sites and M ion occupy the regular sites of FCC structure. This is the general structure of calcium fluoride as well, where fluorine has occupied the tetrahedral interstitial space and calcium has occupied FCC structure. The other properties are listed here in this slide. The synthesis of samples were done by combustion method, and the reaction equation was as shown in this equation. Calcium nitrate and 2 molecules of ammonium fluoride give us the calcium fluoride and nitrous oxide and water. The precursors used were calcium nitrate, ammonium fluoride, dopant manganese nitrate, and urea as a fuel and distilled water. The steps were we took the weight of the precursors according to stoichiometric ratio and add it into the beaker with urea, and for doping we added manganese nitrate according to the doping concentration. Then put on the magnetic stirrer for 30 minutes at 30 degrees Celsius of temperature till the solution form a precipitate. Then that precipitate took into the furnace, and at 400 degrees Celsius we ignite the solution for a few seconds.
Come to your result.
Then the furnace.
Mohini, come to your result directly.
Okay, ma'am. The XRD results are here. As we can see, the peaks coincides with the JCPDS data, and the peaks are shown here, and the HKL values for those are mentioned in the figure. We calculated the from the Scherer equation the crystalline size. This is the WH analysis for the pure calcium fluoride and the doped concentration. This is the summarized table for the crystallite size from WH method and the strain. This is the graph shown for crystallite size with respect to doping concentration and the strain variation. The red line shows the strain variation, the blue line shows the crystalline size. This is the band gap calculated from the UVDRS. This came out to be 5.96, which aligned with the literature values of calcium fluoride band gap. This is the SEM EDS images for calcium fluoride. First one is the calcium fluoride pure. After that, these are doping of manganese SEM images, and this is with increase of doping. We see the some surface charge clusters in the images, and this is the EDS pictures to confirm the elemental composition of the samples. Thank you.
Am I audible?
Yes.
Okay, Mohini, what is the application of your work?
Ma'am, we are going to use these samples for thermoluminescence and photoluminescence characterization, and we are going to find some dosimetric properties if this is feasible for it or not.
Okay.
Thank you. Next, we have Sukhpreet Kaur.
Thank you.
Next up, I would call Vijay Kumar.
Yes, ma'am.
Please present.
Ma'am, is the screen visual?
Yes, it is.
Good morning, everyone. Myself, Vijay Kumar, MSc student from Chandigarh University. I have done the research work on grid hydrothermal synthesis of zinc oxide nanoparticle from Aglaonema plant extract under the supervision of Dr. Sanjeev Kumar. I have done the research for the antibiotic pollution and breast cancer burden. I am using my nanoparticle to degrade the ciprofloxacin dye, which is found across 104 countries throughout the world, and for the breast cancer anti-cancer activity against MCF7 cells, which is generally cause for the breast cancer in the women. It is highest reason of the cancer in the world currently in the world, having case of 2.3 million new cases every year. Next, why we are using ZNO? ZNO is a wide bandgap semiconductor with the bandgap of 3.3 electron volt. Next, why green synthesis? For the chemical synthesis, we get the byproduct which are harmful for the environment. As we are trying to move toward the sustainable future, we are using green synthesis to reduce those byproducts like hydrazine, NaBH4, and many other byproducts. Next, why only Aglaonema? We are generally using the different plants for the different researches. Aglaonema is most widely available in the Punjab region from where I belong, and is not done before, is not used before for any ZNO, copper oxide, or any metal oxide nanoparticles. We are using green hydrothermal synthesis workflow. First, we collected the Aglaonema plants, then we extracted the plant extract. After mixing that with the zinc acetate, we set the pH at 8. After hydrothermal crystallization, we calcine the sample and characterize that with XRD, BET, SEM, and many other characterization. We prepared the 3 samples. One is with plant extract, another only with the distilled water. There is no extract used. And third is industrial used bulk ZNO. For the phytochemical identification, we use the GCMS characterization. In our plant extract, we use the GCMS characterization from where we get there are many dominating phytochemicals in the plant extract. Some of important phytochemicals are 1-monolinoglycol, which is used for capping and size control. Another one is decosinamide, which is used for the initiation of the reaction. From the XRD, we calculate the crystallite size, strain, and lattice parameter of all 3 samples. We got the crystallite size around 29.3 nanometer for our sample. As you can see, in uncalcined sample, there are many secondary peaks. But when we calcine the sample, we got the pure phase ZNO, hexagonal phase ZNO, with the microstrain of 1.7 10 to the power -3 tensile strain. Next, we did the surface morphology using FESM and HRTM characterization of the sample. From the FESM, we got the shape of the particle is around spherical. From the EDS spectrum, we got the elemental composition of 50-50% for ZNO and Zn and oxygen. From the HRTM, we got the particles average size of 26 nanometer, which is approximately similar to the XRD, which approximately similar to XRD, and with the d-spacing of 0.28 nanometer, corresponding to the plane 100 plane. Next, for the optical and surface properties, we did the UV visual, wet isotherm, and zeta potential. From the UV, we have seen the blue shift. As we decrease the size of ZN nanoparticle, the band gap is increased. From the wet isotherm, we got the surface area of 78.4 meter square per gram and the pore diameter of 3.84 nanometer. Similarly, from the zeta potential, we got the point of zero charge at 6.13 pH. To establish the surface functionalization of the nanoparticle, we did the FTIR of plant extract and nanoparticle also. From that nanoparticle, we get the peaks at OH stretching, C double bond O stretching, CO stretching, which established that the phytochemical from the plant extract is capped around the
1 minute
particle.
left.
Yes, ma'am.
You have 1 minute left.
Okay. Next, we did the phytochemical degradation of ciprofloxacin. We got the 95 degradation for the nanoparticle, 68 for uncapped particles. And when we do not use any nanoparticle or any light, we get the 4% degradation and 12% degradation. The most dominating ROS species is OH, and then we have H plus, electron, and O2 negative. After 5 cycles, we get the 86% degradation even after 5 cycles. Next, we did the anti-cancer activity. We did the in vitro analysis. We did the cell cycle and protein cycle. From the in vitro cancer activities, we get the IC50 of 56.8 microgram per milliliter for the ANZ for our nanoparticle. Comparing to literature, one of the literature shown 58 microgram per milliliter, another one showing 72 microgram per liter, which is approximately similar to our literature work. What is the conclusion? We are the first to use aglaonema plant extract for the ZNO synthesis. We got the nanoparticle of 29.3 nanometer. Selectivity index is around 2.42. IC50 is 56.8, and we got the high bed surface area of 78.4 meters square per gram. What are future work? We are trying to use these same nanoparticles for solar-driven photocatalysis, real wastewater matrices, and in vitro study of anti-cancer activity. And different from the different plant, we are trying to synthesize different nanoparticle of metal oxides. Thank you, everyone. Any questions?
Hello. Okay. How
Yes,
you
ma'am.
will be using these nanoparticles for detection of the cancer cell or activities?
No, ma'am, we are not detecting the cancer cell. We are doing the anti-cancer activities. We are already synthesized the cancer cells in the laboratory. We are trying to kill those cancer cells. We are using the apoptosis, ma'am, morphological apoptosis here. These are the morphological.
Have you checked? Have you done any experiment or something
Yes, ma'am.
right on actual situation?
Yes, ma'am. We are doing the MTTSA, ma'am, for the cancer cells in the laboratory, not the actual cases, but in the laboratory, we are doing the MTTSA.
Okay. Do you
Because
have
we
any
are,
plan to collaborate with some hospitals or some?
no, ma'am, not right now. We are trying to look forward for that. Not right now. We are not collaborating without any hospital. Currently, we are doing this in our laboratory.
Okay. Okay, Vijay.
Thank you, ma'am.
Thank you, sir. Next, we would call Kritika Garg.
Good morning, ma'am. Good morning, one and all present here.
Kindly share your presentation.
Yes, ma'am. Just a minute. Ma'am, it is visible.
Yes, please start.
Good morning, everyone. My name is Kritika Garg. I'm research scholar in Manipal University, Jaipur. I'm doing my PhD under guidance of Dr. Devanjan Bhattacharjee. And my topic for today's presentation is structural and vibrational dynamics of fluorinated skip phase liquid crystal and analytical discourse via DFT. So, this is the title of my presentation. In this picture, we can see the image. This is the image of the liquid crystalline samples. So first we will know about the what is liquid crystalline samples and what are the properties of them. So this is the table of content. In this I will explain the introduction about liquid crystals and the theoretical techniques which I have used, and the Raman and IR spectra of the sample, and the conclusion and some references for the study. So first we'll discuss about what is liquid crystal, what is the history of liquid crystal. So in 1888, Friedrich Reinteger observed a liquid crystalline phase in cholesterol benzoate. He melted the cholesterol benzoate. First, he got at 145.5 degrees centigrade, he got a cloudy liquid phase before turning into the liquid. Generally, when we melt a solid structure, it will melt into the liquid state. It will melt into the liquid, but for the cholesterol benzoate, he saw that it will not directly melted into the liquid. It just changes it. It shows an intermediate phase at 145.5 degrees centigrade. Then he concluded, then he gave this research to his physicist friend Otto Lemon, and he concluded this intermediate phase at a new phase of matter, which is called liquid crystalline phase. So as we can see in the diagram that this is the perfectly ordered phase, which is called crystalline phase. Then after increasing the temperature at 145.5 degrees centigrade, the molecules started to align themselves in a different direction, or disordered phase. Then after increasing more temperature, the molecules will be perfectly disordered. This is the isotropic liquid phase. So what is the liquid crystal? What are the types of liquid crystal? So liquid crystals are two types as a thermotropic liquid crystal and leotropic liquid crystal. Basically, thermotropic liquid crystal is based on thermal conditions or temperature dependent studies, and leotropic is based on solution dependent. So and later on, thermotropic classified into two types of liquid crystals, that calamatic and discotic liquid crystals. Then after that, discotic is classified into the discotic nematic and discotic columnar. So my study is based on the thermotropic calamatic liquid crystals. That is calamatic after classified into the three phases: nematic, smectic, and cholesteric. So in this study, I will be discussing about the 507 liquid crystal. That is a nematic liquid crystal. So what is nematic means? Nematic, basically nematic words originate from a Greek word nema, which means thread-like structure. So in the nematic stage, molecules start aligned to a specific direction. As we can see in the next slide, that this is the calamatic phase of the liquid crystal. In the calamatic phase, the molecules are rod-like structure. Molecule shows rod-like structure. Then after increasing temperature, they shows a short range orientational order. They are not perfectly ordered and not perfectly disordered, but they show short range order. Then after increasing more temperature, some parallel alignment, they start to align in the parallel direction. That as we can see that they shows a orientational long range order. After increasing more temperature, the symmetry will be reduced, and they start forming a layer which is called smectic phase. In the smectic phase, the molecules are in orientation, shows orientational and positional long range order, but in a nematic phase, the molecule shows only orientational long range order.
Ma'am, you have 1 minute left.
Okay. So 507 is like NOM series liquid crystal. And this is the diagram of NOM series liquid crystal, where N and M represent the number of carbon atoms, and N's double bond CH is a skiff base. So I have used DFT, density functional theory, in this study. So this is the optimized structure which I have got from DFT using Gaussian 09 software with the 631G plus DP software. These are the fluorines which are connected to the benzene ring. And this is the phase transition temperature, as we discussed, as I discussed earlier, that after increasing temperature, it changes its phases as smectic. Then after increasing more temperature, it changes its phase is nematic, and then isotropic liquid phase. So this is the Raman spectra which I have got from DFT. As we can see in the spectra that at 1627 degrees, 1677 centimeter inverse, this is This is due to the C double bond and stretching. That C double bond and bond present in the structure, and also this is the bond 1133 and 943 940. It is because of CF bond, which is mainly concerned in this study, the effect of fluorine. What is the effect of fluorine on the 507 liquid crystalline compound? So this highlights the significance and influence of the fluorine substitution. And this is the IR spectroscopy. For IR spectroscopy, as it confirmed the functional groups present in the system, and also it validates the molecular structure and fluorine incorporation in the liquid crystal. So with this study, we have got that that fluorine substitution influence the molecular vibrations, electronic distribution, and intermolecular interactions also. And DFT analysis revealed that the molecule after incorporation of fluorine, the molecule is stable, and it shows a structural anisotropy, which is the main characteristics of liquid crystal. And also this finding offers a strong foundation for designing new advanced liquid crystalline materials, which will be used for electro-optical and display application. These are the references. I have published some papers regarding the skip-based liquid crystals. So thank you so much. This is my presentation.
Yeah, Kritika,
Yeah.
which exchange correlation you have used within density functional theory?
Sorry, ma'am, I couldn't hear you.
Which exchange correlation you have used for DFT?
B3LYP functional.
Why so?
My B3LYP functional gives a clear understanding, and after reviewing the literature, it shows us some better results comparison to other, which is CAM, B3LYP, and other exchange correlation functions.
Okay, Kritika, you may leave.
Okay, thank you so much, ma'am. Thank you.
Thank you. Next, I would call Shivani Mishra.
I am audible, ma'am?
Yes, you're audible. Please present.
Good morning, everyone. My name is Shivani Mishra. I am a research scholar in Manipal University, Jaipur. I am doing my PhD under the supervision of Dr. Kishore Kumar Shah. I presenting my topic on the investigation of structural and optical response of semi-metallic oxide-based perovskite Ba2FeMnO6 using density functional theory. And this is the outline of my presentation. And first of all, my introduction. Ba2FeMnO6 is double perovskite material where two different transition metal, iron Fe and manganese Mn, are present. This makes the material highly stable and gives its unique electronic and magnetic properties. These materials are widely used in electronic sensor, magnetic storage device, solar cell. It can convert sunlight into electricity and also detect magnetic temperature charges. So studying its property is important for advanced electronic and spintronic applications. And this is the computational methodology. In this work, I use density functional theory, and method is FPLAPW, full potential linearized augmented plane wave, and software is Win2K. Functional is a GGA PBE. Spin polarized calculation were used to study magnetic behavior. Structure was optimized to find the most stable configuration. This approach helps calculated electronic, magnetic, and properties accurately. And this is the crystal structure of this material. The material has a cubic double perovskite structure. It belongs to the FM bar 3M space space group which is highly symmetric in this structure, and the BA occupy corner position. Fe and Mn are at specific lattice side. Oxygen form octahedral coordination. This structure is responsible for its stability and properties. And this is the band structure of the material. Band structure is so how electron energy change with momentum k points in my graph. The x-axis represent k point k. K points is highly symmetric points, and the y-axis represent energy electron volt. The horizontal line at 0 electron volt is the Fermi level. Now looking at my result in the spin up band cross the Fermi level, so it is behave like a metal. Behave like a metal, and spin down there is a small band gap. A band gap is a 0.35 electron volt. So it behaves like a semiconductor. So overall the material is half metallic, and this is the density of state. Density of state shows how many electronic states are available at each energy level. X-axis energy electron volt, and y-axis
We have
is
1 minute left.
in my graph. Spin up does has stable at Fermi level metallic behavior, and spin down so as a gap. So this is a semiconducting behavior. Also uh, we see P and D hybridization, and Fe is d orbital. D orbital, this interaction control conductivity and magnetism. And this is the optical properties. Optical property describe how two material interact with light. We analyzed parameter like absorption coefficient, reflectivity, refractive index. This absorption coefficient is also which this absorption coefficient is low, which means the material does not absorb light, and the x-axis so energy
Please conclude.
electron volt, and y-axis so optical parameter, and x absorption reflectivity. Here we see low values and low interaction with light, so it is less suitable for solar cell, uh, but the but they are better for magnetic device spintronic applications. And this is the result and discussion from the study. Structure is stable cubic double perovskite, and material is uh, half metallic, and band gap spin down is 0.35 electron volt. Total magnetic moment is 6 mu b, and Fe contribution 3.52 mu b, and Min contribution 2.448 mu b. Thank you, ma'am.
Hello, Shivani. Why you have chosen the PBE exchange potential, as it is well known to underestimate the band gap of the materials?
Sorry, ma'am.
You have performed your calculation with the help of PBE, right?
Yes.
But PBE is known to underestimate the band gap of the material. Why you have chosen it?
We also choose ma'am is a MBJ for calculation.
How much difference you are getting in PB and MBJ?
Sorry, ma'am.
How much difference you are getting in PB and MBJ?
Ma'am, for MBJ running.
Okay. Okay, Shivani. You may leave.
Thank you, ma'am.
Thank you. Next, I would call Yogita Bansal.
Hello, hello, hello.
Yes, you're audible.
Am I audible? Um, Actually, I have lost. Are you able to see it?
Shivani, can you please stop sharing your screen? Yogita, you may share now.
Is my screen fully visible? I think some portions are not clear. Hello?
Your screen is visible. You may start.
Okay. Good morning, all the teachers and all the fellow research scholars. My name is Yogita. I'm in the 5th year of my PhD, which I'm pursuing in bio-nanotechnology. So the topic of my research is to synthesize phytogel-like semi-synthetic biomaterials which can mimic brain extracellular matrix. As we all know, a brain is the most important part of our body and also a vital part of body. It is extremely important because it is the controller. Um, but it is extremely soft at the same time. So it is protected by a very hard shell, which is known as skull. Um, Despite being protected by a very hard covering, it due to neurological disorders, accidents, and traumas, the brain tissue is lost. So, but that's not the only problem because other tissues maybe can be repaired easily. But the problem with brain ECM, brain tissue, is that once it is lost, it is very hard to regenerate it because the brain cells are terminally differentiated, and and also many of the physiological pathways are still unknown. So because of all those vague mechanisms, the repair process is extremely difficult. So but my focus is not traumas or ballistic damage. My focus is the disorder which is known as glioblastoma. It is a type of cancer which occurs in the glial cells. So because of this cancer also, our brain gets damaged very adversely. It is a grade 4th cancer. So in order to study various mechanisms of glioblastoma, I synthesized hydrogels, which are the biomaterials which can mimic the brain extracellular matrix. So previously, 2D and 3D cultures, phantoms were used. But they because they don't have stimulus responsiveness. So nowadays, 4D are being used. So in order to synthesize, I used gorgum and acrylic acid, and crosslinker was leucine. So these are the results. If we see the FTIR result, um, in gorgum, I didn't show the peak at 3,427. There is a prominent peak. But after the grafting. The method was graft copolymerization to synthesize this hydrogel. So after I'm only showing the results after hydrogel synthesis. So I had two. I synthesized two variants: phytogel 1 and phytogel 2. So if we look at the results, the peak at 3427 disappeared, which confirmed the hydrogel synthesis. For the TG analysis, it shows three-step degradation, and there is the percent decomposition is the same, which means there is no filler. So which I didn't use. XRD, if we see, I didn't use the neat gorgum. It has a very prominent, it has somewhat crystalline structure, but here it does not have two peaks. If we look at the SEM results, some nanometer, 17 nanometers pores were
Ma'am, you have 1
observed.
minute left.
Okay, these are the this is the EDEX analysis. The percentage composition is written here. All the expected elements were detected, except in one. Nitrogen was not detected because low detection limit, and Raman spectra is flat because of too much hydration level. These are the most important results, which rheology results, which I mainly focused. This is amplitude sweep. If we compare it, this is used for defining linear viscoelastic region. So if we compare it with porcine brain, porcine is like pig brain, which is very similar to human brains and usually used for brain analysis. So it is quite matching with the porcine, because the LVR is near about 70 and ours is near about 100. This is amplitude or frequency sweep result, and this is thixotropy, which is very important. This is the reason why we use biomaterials, because they have the ability to bounce back to the original structure. So all my results
Yogita,
are in line with the human
Yogita,
like brain. Thank you very much.
what is the USP of your work?
USP means?
What is special in your work or something that has not been done and you have done?
Rheology. These are these three results: thixotrophy, amplitude sweep, and frequency sweep. These are extremely important because it is biophysics, not normal biology. Because here we look at the viscoelastic properties. It is a new field actually. These are the results which confirm whether the culture will degrade or not. Because if the rheology is not matching with the human brain, then culture is destroyed because graft rejection occurs. Because human brain has a specific viscoelasticity. If it won't match, then we can't proceed any further. So my results are matching with somewhat matching with the human brain.
Okay, okay, Yogita.
And this is extremely new topic, very rare in India.
Thank you. Next, I
Thank
will
you.
call Anju Rani.
Straight to wind up in 5 minutes. So better to come to your result directly rather than providing the introduction and reading out the slide.
Anju Rani, are you there? Next, we have Malika Redu. Ma'am, you're not audible to us. Miss Anju, you're not audible to us. Ms. Sanju, you can check with your mic. I would call the next presenter, Mr. Om Prakash Yadav.
Hello, ma'am. I am audible.
Yes, you're audible. Please present.
My screen is visible, ma'am.
Yes, please start.
Okay. Thank you. Hello, everyone. My name is Om Prakash Yadav. I am research scholar in Department of Physics, Manipal University, Jaipur. I am doing my PhD under the supervision of Dr. Kishore Kumar sir. Today, my PPT topic is effect of antimony substitutions on structural and optical properties of RB2 BIGACL6 perovskite using density functional theory. These are my outlines of today PPT. In introduction, we work on halide double perovskite. The chemical formula of halide double perovskite is A2BP-X6. Here, A is metal alkynes. These are large cations. B and B dash is two different cations. There are metal cations and X is anions. Halide double perovskite are emerging as lead-free alternative for optoelectronic application due to their environmental stability and tunable properties. Here, we substituted 50% antimony at the side of Pi in RB2BiGaCl6 and detailed analysis of band structure, density of state, absorption coefficient, reflectivity, refraction index using LAPW method. In computational methodology, we use density functional theory framework. The method is used full potential linearized equivalent plane wave methods. We work on Win2K software, and in exchange correlation potential, we use GGA PP approximations. Here we calculate non-spin polarization and K points is 6 into 6 into 6. This is crystal structure of pure material and doped material. We doped 50% antimony, and there is the positions of atoms in the crystal structure. Here crystal structure is cubic and space group is FM3 bar M. In band analysis, we found band and dose graph for pure material and doped material. In pure material, x-axis represent K points and for dose density of state, and y-axis represent energy in electron volt. In pure material, the direct band gap is 0.892 electron volt. In doped materials, band gap is reduced approximate 52%, and which is 0.472 electron volt. Here, density of states also known so that there is no state are present at Fermi level, which is confirmed that material is a semiconductor.
So you have 1 minute left. Please conclude in that.
This is my partial density of states graphs, and here dominate Cl p and orbital and bismuth p orbital, and antimony p orbital is given major role in optical properties. Here we calculate absorption coefficient, reflectivity, and reflective index. In visible region, we saw the absorption is increased in doped material, and IR region increase absorption in doped material, and visible region and UV region absorptions are slightly changed in absorption observed. Reflectivity is
Sir, please
visible
continue.
region slightly increase, and reflective index is maintain strong interactions. These are results we have discussed in graphs. These are references we used. And thank you, ma'am.
Om Prakash, why you
Yes,
have
ma'am.
chosen the antimony for the doping?
Ma'am, antimony सब्स्टिट्यूट करके देखा है कि क्या चेंजेस आएगा इसमें एक बार.
But what additional or some drastic change you are observing that is not shown here? It is reducing from 0.8 to 0.4. So what kind of the application or the promising potential you are observing in the doping?
Sorry, ma'am, I'm not understanding.
आपने एसबी को डोप किया है, ओके?
Yes,
आपका
ma'am.
बैंड गैप 0.8 से 0.4 पर रिड्यूस हुआ है. उससे ऐसा
Yeah.
क्या ड्रास्टिक या बहुत ज्यादा चेंज आ गया कि आपको लगता है कि आपको एसबी को डोप करना चाहिए था?
मैम, मैं अदर अदर डोपिंग लेके देख रहे हैं अभी कि कौन सा डोप ज्यादा अच्छा रहेगा.
ओके ओके.
Thank you. I would request Anju Rani to now present. Anju Rani, please present.
Hello, ma'am, am I audible?
Yes, you're audible.
Mhm.
Please share your screen.
Yes, ma'am, I'm sharing. Actually, ma'am, I'm presenting from my laptop because there is some problem in my problem in my laptop, so presenting from phone.
That's fine, but please do it ASAP.
Yes, ma'am. Ma'am, just give me 1 minute.
Okay.
Actually, ma'am, I'm not able to share my screen.
Then we would move on to the next participant.
Okay, ma'am.
Is Ishita Kapil present?
Yes, ma'am.
Please present.
Okay, ma'am. Is my screen visible, ma'am?
Yes.
Hello.
Your screen is visible. Please start.
Okay. Greetings of the day to everyone present here. Myself, Ishita Kapil, a PhD scholar working under the guidance of Dr. Haryana Bhadari, Department of Physics, Amity School of Applied Sciences, Amity University, Haryana, Gurugram, India. So the topic of my today's presentation is photocatalytic study of mixed phase manganese oxide nanoparticles. The content listed in this presentation includes the introduction, motivation, synthesis process, results and discussion, photocatalytic activity, conclusion, references, and acknowledgement. So, beginning with the motivation. The prime motive for studying the photocatalytic activity of the prepared sample was to make sure that the availability of the water should be based both on quantity and quality, because as we know nowadays what happens that organic dyes are the well-known source of contamination as they are toxic, leads to cancer causing and threaten human and aquatic life. Moreover, if we use the traditional wastewater treatments, they often fall short, grappling with the issues like cost or inherent complexities. So the main motive of performing photocatalytic activity is to overcome with all these problems as much as it is possible. So there are various applications of metal oxide nanoparticles like in catalysis, sensors, wastewater remediations, biomedical, batteries, solar cells. So the main motive, so that's why metal oxide nanoparticles are considered to be the most promising catalyst for degrading dyes in a facial way, as reason being they have a high surface area to volume ratio, because of which they easily break down the dyes into a non-toxic products by producing hydroxyl radicals under the light exposures. Next slide comparisons there. So why there is a need, why I have chosen manganese oxide. So because of its various properties, like it has a tunable band gap, easy to work with, has a high redox potential, nanostructures offer abundant reactions, stable and robust. Moreover, it can be produced in abundance and is also non-toxic in nature. I'm skipping this synthesis process due to a shortage of time. Next comes the results and a discussion part where I've performed the characterizations like XRD, Raman spectroscopy, FTIR, UV visible, TGA, DSC, and further I've studied photocatalytic activity. Beginning with the X-ray diffraction pattern. So figure 1 shows the XRD pattern of as synthesized mixed phase manganese oxides. Means basically they are the samples as prepared, they are not annealed in figure 1. And figure 2 shows the XRD pattern of the synthesized mixed phase manganese oxide nanoparticles annealed using with and without surfactants. So basically during the synthesis process, I've used 3 surfactants, that is SDS, PEG and KCl, and 1 sample. So total 4 samples are prepared, 3 using a surfactant and 1 was without a surfactant. So the XRD pattern shows the formation of amorphous alpha MnO2 and alpha MnO3 phases. The data matches well with the JCPD's numbers as mentioned in the text, and the XRD peaks of the annealed sample shows shows mixed MnO2 and Mn2O3 phases with high crystallinity. Moreover, we have calculated the crystallized size using the Scherer equation for this individually for the alpha MnO2 phase of the materials and the for the alpha Mn2O3 phases of the nanomaterials.
You have
Next
1
slide
minute left.
comprises of the Raman spectroscopy. So basically all prepared samples have shown a strong MnO2 band nearly at around 629 to 637 centimeter inverse, confirming MnO6 vibrations further, a strong shoulder peaks around 5-6 in a range of 562 to 587 appears, indicating the presence of Mn2O3. Moreover, a broad peak suggests the lattice disorder confinement. So, in case of MO SDS and MO KCl, it basically exhibits a stronger MnO2 signatures, while while the mixed phase manganese oxide nanoparticles prepared without surfactant, they show a Mn2O3 feature, which highlights the surfactant influence on the phase evolution. And as XRD confirms the formation of mixed phases like MnO2 and MnO3. Similarly, Raman spectra has also confirmed the mixed phase, enhancing the MnO2 signature and reducing the Mn2O3 dominance.
Ma'am, your
Next.
time's up. Please conclude.
Okay. Next, the TG and DSC profile of MnO2. So basically, we have performed the thermogravimetric analysis, where there are basically the three stages at the temperature up to 220 degrees Celsius. It was a first stage, which was for the removal of surface water. Then in a range of 230 to 450 degrees Celsius, major decomposition has taken place, and a phase transition from alpha Mn2O3 to alpha Mn3O4, it was taken around 460 to 780 degrees Celsius. So in case of MOSDS, it shows the lowest mass loss of around 3.5%, which in comparison to the sample prepared without using a surfactant. For that sample, it was around 24.3%. In case of using KCl as a surfactant, it was 19.8%, and for MOPEG, it was 15.7%, which was highlighting the superior thermal stability of the prepared samples. Moreover, in case of SGS shows the strongest, act as the strongest stabilizing
You need to
agent.
stop now.
Hello?
You need to stop.
Hello?
Your presentation time is over now. Please please stop the presentation. Thank you.
What is the novelty of your work?
Ma'am, so basically what we have done, we have used, the first novelty is that we prepared using a surfactant of, and basically we have used a very minimal amount of surfactant, but while we have, I can show in the slide that in a photocatalytic performance, we have got a remarkable results. Like we can see that when we have the samples prepared using without surfactant has shown 79.2% of the degradation in 150 minutes. Moreover, the samples prepared using PEG and SDS KCl, they have shown approximately in a range of 80 to 84.4%. So, the sample prepared using KCl has shown a better result, even with a minimal amount of addition. It was like 84.4%. So, yeah, basically, I could say that these surfactants have enhances our photocatalytic performance.
Okay.
Thank you, ma'am.
Next, I would call Malika Reddy.
Ma'am, shall I present now? Anju Rani, ma'am.
If you are able to, sure.
Yes, ma'am, sure. Just
Yes,
wait a
ma'am.
second, present my presentation.
Ma'am, I'm sharing.
Malika, can you
I
just
share,
wait?
ma'am?
You can present next.
Can I share?
Yes, ma'am, sure.
Yes, you can share. You have 5 minutes. At 5 minutes, we'll cut you off there.
Ma'am, is it visible?
Yes, your screen is visible. Anju, you may present after this.
Okay. First of all, I would like to say good afternoon, everyone. I am Malika Reddy from the Department of Physics, University Institute of Science, Chandigarh University. Today, I am going to present my topic, bioengineered SNO2 nanoparticles for pollutant degradation and microbial control. Basically, this work focuses on the potential of bioengineered SNO2 nanomaterials as a functional platform for sustainable water remediation. Then before moving ahead, here is the contents of my presentation. First, I will briefly discuss the water contamination challenge. Then I will explain why tin oxide is important and why bioengineer synthesis is valuable.
Malika, go
After
ahead.
that, I will Hello?
Malika, come to your work.
Yes. Hello?
You only have 4 minutes, so please do fast.
I'm audible?
Yes, you're audible. Please continue.
Okay. Then I will explain why tin oxide is
The
important
judges are
and
asking
why
for you to just show your
bioengineer
work
synthesis
directly.
is valuable. After that, I will discuss the structure-property relationship followed by the mechanisms of the
pollutant degradation and microbial control. And then finally, I will highlight the sustainable development, key challenges, and my overall conclusion. Basically, water contamination is a serious global issue, and it is often a dual challenge rather than a single one for us. On one side, we have persistent organic pollutants, which are highly
Malika?
stable and very difficult to remove
Malika?
using conventional methods.
Malika, please could you skip the introduction and go directly to your work? Your solution.
So you are not audible to me.
Can you please directly skip to your work since you have very less time with you?
Okay. And basically, I have started my PPT, and then this is the basically brief discussion what I'm going to present. And this is the aim of this presentation. The first aim of this presentation is to explain why bioengineered tin oxide nanoparticles are promising for vapor remediation. The second aim is to show how sustainable material design can improve pollutant degradation performance. And the third aim is to connect the nanoscale properties of tin oxide with its microbial control mechanism. So overall, this presentation tries to link the green synthesis, material functionality, and experimental work in a single framework. Then the question arises why we are using SNO2. Since tin oxide is an attractive material just because of its high chemical stability. This makes it suitable for use in our aqueous and environmental conditions. It also has active surface reactivity, which helps in the adsorption and surface mediated reactions. And the third one, another important feature is its defect sensitive electronic structure. Particularly in this, we have oxygen vacancies and defects that can influence charge transfer behavior, which is very important in our catalytic processes. Since it also shows antimicrobial relevance, that means it's just not only useful for the pollutant removal, but also for our microbial control. Then the question arises why we are using just only bioengineered synthesis. This is not important only because it is greener than the chemical method, but also because it can affect the final material properties. It also reduces the use of harsh chemical reagents and helps minimize toxic generation. At the same time, biological molecules can influence nucleation, particle growth, surface chemistry during the formation. This means the synthesis route itself can help tune the surface and interfacial properties. So the advantage is actually twofold. Firstly, it also supports sustainability, and second, it can improve the functional performance. That is why bioengineered synthesis is especially relevant for the environmental nanomaterials. Then we have its structural property featuring governed performance. The performance of SNO2 nanoparticle is property driven. It is not governed only by its chemical composition, but also by the nanoscale structure and surface features. For example, surface area and active site influences how effectively pollutants can interact with the nanomaterials. Surface functional groups affect adsorption and interfacial behavior. Then we have also defects and oxygen vacancy states that are especially important because they influence charge transfer and the ROS. These same features also affect the nanoparticles interacting with the microbial cells. So overall, the remediation of SNO2 depends strongly on the relationship between its structure, surface chemistry, defects, and functionality. Then we have the mechanism of pollutant degradation. The process begins with activation of the tin oxide nanoparticle surface. This leads to charge separation, means electrons and holes are separated, or we can say generated. As a result, the ROS are formed. These are the reactive species that attacks the pollutant molecules adsorbed on the SNO2 surface.
Ma'am, you have 1 minute left. Please conclude.
Yes, ma'am.
You have 1 minute left. Please conclude.
Yeah, through continuous oxidation and the related surface reactions, hazardous contaminants and broken down into simpler and less harmful products. Then the last one is we have the mechanism of microbial control. First is its surface contact where nanoparticles interact with microbial cell membranes. Second is oxidative stress. And the third one is growth inhibition, which where important biological processes are impaired. So unlike materials that perform only catalytic degradation, tin oxide has the advantage of also contributing to antimicrobial actions. This makes it highly relevant for water system where both chemical and microbial contaminants must be addressed together. And in the end, I would like to show by the key challenges. The first is lab synthesis, where morphology and composition still needs to optimized more precisely. Second one is mechanistic clarity, because the exact pathways, the roles of defects are not yet fully understood. The third one is stability testing. The fourth is performance in real water, because materials that work in laboratory may behave differently when they are in aqueous environments. And finally, we have a challenge of for scale up, which is necessary for particular development. Then I would like to conclude it with that SNO2 emerges as a promising multifunctional nanomaterial for environment remediation. It shows strong potential for both pollutant degradation and microbial control. Because of this dual function behavior, tin oxide represents a green and promising platform for the sustainable water remediation technologies. And thank you for your attention. I would happy.
What is your future plan to check the feasibility of these nanoparticle for the water applications?
And basically for the water applications, the work is this important because SNO2 has broader applications since in catalysis, but my focus is just relevance to aqueous remediation there.
Okay.
Thank you. Do we have Shivam Kumar present?
Can Shivam Kumar show the presentation from the same laptop and from the same ID?
If he is present, then sure. Anjurwani, are you ready to present?
Yes, ma'am.
Yes, ma'am. Please be ready to present next and make sure you're able to show your presentation.
Okay, ma'am.
Shivam Kumar, please start. And please switch directly to your work and not the introduction.
Hello, good evening, ma'am. I'm audible?
Yes, you're audible. Please start.
Yeah. So my work on the green synthesis of nickel oxide nanomaterials by the plant extract of Dracana compacta. So my main
Could you
work.
please skip the introduction and go directly to your work?
Hello. Yes, sir. Your is not audible. What are you talking about?
Can you please skip the introduction and go directly to your work?
Okay, sure, ma'am, sure. So basically, my work on the green synthesis of nickel oxide nanomaterials. So our approach is here. We are using the green synthesis method here for the preparation of nanoparticles. So I mean, work on the basically green synthesis. The traditional chemical synthesis method using mostly using a toxic chemicals and the harmful solvents, which is harmful for the environment also. To solve this problem, we use a plant extract here basically. So main R is basically we are working on a plant extract of Dracana compacta. So the research gap is here. No systematic study was take place on a Dracana compactor right now, and we are also working on the basis of the phytochemicals present in the Dracana compactor to prepare the nickel oxide nanomaterials, and how the precursor concentration affects the crystallinity of the material, and how we can calculate the band gap, and some also anti-cancer, antibacterial properties was there. And also there's not such kind of information about XPS and PET analysis in the case of nickel oxide nanomaterials with the help of Dracana compacta. So we are going to work here basically. So we can see the some pictures are given to you, like I have taken the plant from the Muskan nursery, New White Arts, and we have to make a plant extract from the sexualate apparatus, and we use a salt here, that is a nickel chloride, hexahydrate, and we prepare this basically the particles in the three different concentration to measure the essential effects like 0.1, 0.4, and 0.8. Also, we are maintaining the pH here for using 3 or 4 drops of ammonia, and we use autoclave for maintaining the temperature of 160 degrees Celsius. And at the end, we purify the solution and then make a nickel oxide nanomaterials. So after that, we are basically done the LCMS, which identify the phytochemical present in inside the plant extract. Basically, what kind of major phytochemicals are there. So you can see the Si are negative, that basically the ions uh, that plant extract have. The major kind of phytochemicals we have detected, that is a gallic acid, caffeinic acid, cetirizines, and some quinine, which is essential to control the particle size in the formation of nickel oxide nanomaterials. So we have used the different three concentrations, 0.1, 0.4, and 0.8. What we have studied here, basically the low concentration precursor, make a larger crystalline size and lower defective density and wider band gap. When we goes to the 0.4 concentration, the intermediate concentration, basically the balanced nucleation take place and the moderate crystallinity is formed, and 0.8 concentration, that is a very higher concentration, the band gap would be 3.70, and the refinement is higher in that case because of nucleation. We also done the XRD of the three different concentrations, 0.1, 0.4, and 0.8, to identify what kind of crystalline size was there. And also we have studied the optical properties, and we have identified the 0.1, 0.4, and 0.8, what kind of the wavelength was take place, and what would be
You
the
have 1
effect of...
minute left. Please conclude.
Sorry, ma'am. Can you speak again?
You have 1 minute left. Please conclude.
Yeah. So the 0.1, that is a 224 nanometer, 0.4, that is 228, and 0.8, 231 nanometer was the band gap we have done, and that is for liquid and solid. We have also identified the solid nickel oxide materials. And we are going to study also the SEM, airtime and bed analysis, which we identify the quasi-spherical morphology will take place in nickel and oxygen. And also we have done the HRTAM, high resolution transmission electron microscopy, and bed for surface analysis. And at the end, we have also studied the anti-cancer, antibacterial, antifungal properties, and that was the IC50 value 3.69 for the very good criteria for the anti-cancer property here. And we can see the further when we increase the concentration of the kind of the nickel oxide nanoparticles, the cancer cells would get completely destroyed with the help of nickel ions are formed, and which attack on the bacterial membranes or the cell membranes basically. So this is a whole presentation of mine.
Hello. Shiva,
Hello.
which
Yeah, ma'am.
plant you have taken for the synthesis of the NiO2 particles?
Ma'am, we have taken the Dracana compacta, which is used in houses for decoration actually.
And why have you have chosen only this?
Because, ma'am, there is no kind of literature we have found for this plant extract. So that is our work. We have to use this plant extract here.
Is there any report on the synthesis of the NiO2 NiO nanoparticle with the other plants or green synthesis method?
Yeah, ma'am, there's a lot of different kind of plants like in like normally like neem and rosy rows of different flowers, also seeds in even different most of the cases में like जितने भी plants उनका use हो चुका है, so that's why we are using this one.
Okay, thank you. Next, Anjurani, you may present now.
Yeah.
Okay. Shall I share the screen, ma'am?
Yes, please do.
Ma'am, it is showing that you cannot share.
Malika, could you please stop sharing your screen so Anju can share her screen?
Malika and Shivan, please stop sharing your screen.
Okay, sure, ma'am. Thank you, ma'am.
Anju, you can try sharing your screen now.
Yes, sir, I'm trying.
Yeah. She's not there in the meeting.
Is it visible, sir?
Yes, it is.
Good afternoon, everyone. Today, my topic of presentation is influence of series resistance, shunt resistance, and temperature on the performance of CTT-based solar cells. These are the contents for my presentation, which includes introduction, literature review, then research methodology, results and discussion, then conclusion, and finally references. So in introduction part, as we all know that sun is non-vanishing and renewable source of energy, which is free from environmental pollution. And we know that cadmium telluride and cadmium sulfide solar cell is a promising candidate for photovoltaic energy production, as fabrication costs are compared by silicon wafers. So objective here is to analyze the impact of series resistance, shunt resistance, and temperature on the performance of the solar cell using SCAPS simulation software. SCAPS is a one-dimensional solar cell capacitance simulation software, which was developed by Alice University of Chelt, Belgium. Then what is SCAPS? And so in SCAPS, we can optimize various parameters like band gap, thickness, and efficiency, temperature, all everything. And we can just study the impact of these parameters on the performance of the solar cells. And we know that traditionally developing a new solar cell and to make the changes to one of the parameters at a time, which is very time expensive and time consuming. So we use SCAPS in order to make it faster and save time and money. So the key performance parameters for SCAPS here are efficiency, open circuit voltage, short circuit current density, and fill factor. This is some part of literature review in which series resistance, which is causes voltage drop, shunt resistance, which leads to recombination losses, and temperature affects the carrier mobility. So motivation here is that CDT's production hit to 15.5 gigawatt in 2024, and losses from series shunt and resistance and temperature limit the commercialization of the efficiency from 18 to 20%, but we can optimize this efficiency using back contact, buffer optimization, and series shunt resistance and temperature. So application, we all know that it is rooftop PV, utility scale, and various other applications are also there. So in methodology, here we have used the material as CDT, CDS based solar cells, in which we have after the introduction of P plus type thin CDT layer as back surface layer, and this figure 3 showing the SCAPS action panel, in which we have to vary the series resistance, shunt resistance, and temperature to see the effect on the solar cell capacitance simulator. So this is the simulation procedure for SCAPS, in which first of all we launch the SCAP, then we just set the problem and give the input parameters to the problem, and then set working condition, then specify the actions which has to be performed, and finally we run the program. This is my result and discussion part, in which I have just varied the series resistance from 0 to 8 ohm centimeter square, temperature from 230 to 400 Kelvin, and shunt resistance from 0.2 to 1.8 kilo ohm centimeter square. And we have just seen the effect of these variations on the performance of the solar cell. So here the band gaps of the various layers are kept constant, like for CDT 1.5 electron volt, CDS 2.4 electron volt, and then for SNO2 3.6 electron volt. These are the variations of the performance solar cell parameters like VOC, JSC, fill factor, and efficiency by varying the temperature, then shunt series resistance, then shunt resistance. So we can just see that from the graphs, we can say that the impact of temperature as the temperature is a critical environmental factor which influence the operational stability and efficiency of the devices, and the result emphasize that importance of thermal management and material stability in CDT based solar cells. for deployment in high temperature environment. And then from the series resistance graph, which is 5B, we can see that it has negligible effect on open circuit voltage and short circuit current density, but fill factor significantly higher with RS, which is leading to the pronounced reduction in power conversion efficiency. So this decline is arising from the resistive losses and limit current flow and distort the JV curve, thereby reducing the maximum power point. And so to analyze
No,
the
ma'am. Your time
yes,
is
ma'am.
up.
And we have also seen that the in figure 5C, the shunt resistance, that as the shunt resistance is increases, the performance of the solar cell is increases. And these are the impacts of the series and shunt resistance on the JV characteristics of solar cell, which clearly shows that as the series resistance is increasing, it just decline the performance of the solar cell, and as we increase the shunt resistance, the performance of the solar cell is increased. So this is the conclusion part. So as I have already told, that series resistance
Ma'am, your time is up. Please stop.
okay, ma'am. These are the acknowledgement.
Yeah, Anju, I have a basic question to you. CDT belong to the second generation of the solar cells. Right
Yes,
now
ma'am.
we are on the fourth generation. People are working on the perovskite, DSC, etc. Why you have chosen the CDT?
Because ma'am, it is first of all, it is cheaper than that, and SCAPs, which is specially developed for SCAP CDT, and so it works well with CDT also, and there is scope for improvement in CDT solar cells.
But all kind of the properties and the outputs are already known for the CDT. What will be the novelty of your work?
Novelty, ma'am, it is just, I have just in basic CDT solar cell, I have introduced one P plus back surface filled layer, which is very thin. And after the introduction of that layer also, the performance of the solar cell was found to increase, means there is increase in the efficiency of the solar cell. And after the introduction of this P plus layer, I have just studied the variation of parameters with series resistance, shunt resistance, and temperature. And I've done, ma'am, layer engineering also, like the variation of thickness, and then studied its effect on the various performance parameters, and just try to optimize so that we can get more efficiency.
Thank you. Next, I would call Aarti Peswani.
Am I audible, ma'am?
Yes, you are. Please present.
Okay. Ma'am, is this visible?
No. Please share your screen.
Yes, is this visible?
Yes, you're visible now.
Thank you. Okay, good afternoon, everyone. My name is Aarti Peshwani from MNIT Jaipur. Today I'm giving an oral presentation, and I'm going to talk about the important and rapid development of organic compound is known as a CH activation. So the title of my presentation is Sustainable Approach to Fuse Imidazoisoquinolines by the ruthenium catalyzed CH annulation using a calcium carbide. So there are the several contents and I will explain each one by one. So, according to the John Dalton, the organometallic chemistry is the tenseless to endless creative nature where the metal atom dance with organic molecule and to form the beautiful complex. The metal catalysis, so the metal catalysis uses as a metal to make a chemical reaction is faster and efficient. So the metal atom is bonded with carbon. The different nature of carbon is sp3, sp2, and sp hybridized. There are the two type of the metal catalysis chemistry, the main group metal chemistry and the transition metal chemistry. Main group metal chemistry, the metal is bonded is an ionic form. So the carbon nature is the nucleophiles and it is the unstable to air in moisture. And the transition metal chemistry, generally this form the coordination compound. So this is the carbon nature have the nucleophilic and electrophilic and also stable air and moisture. In 1967, Fujiwara et al. Have published the first reaction of the CH activation have done by the palladium catalyst. So the CH activation, the transition metal have the major role in the CH activation. CH activation here the hydrocarbon first to react with the metal catalyst to create an organometallic complex in which the hydrocarbon is coordinated to the inner sphere of metal and the formation of an intermediate alkene or arene complex. So the general properties of the CH activation, there is a no need of pre-functionalization and elements products reduce the number of steps. This is a general mechanism of the CH activation have done by the Fujiwara Moritani. The directing group assisted CH activation. So the directing group have interact with the metal and the formation of the beautiful cyclic intermediate and the given is a new functionalized molecule. The different types of the directing group is nitrogen based, oxygen based and the sulfur based directing groups like pyridine, amides, thiamides. Next is the ortho-CH activation. So the ortho-CH activation, what is the ortho-CH activation? So the ortho-CH activation provide the nearest selective ortho site of the cleavage of the CH bond. The directing group have the major role in ortho-CH activation because directing group bind transition metal to the substrate through the agostic interaction. This is a general mechanism of the directing group with ortho-CH activation. And this is a just example. In 2022, when one of my seniors, Sangeeta Kumari, have published a paper of the ortho-CH activation. This is, I brief explain the general introduction of the calcium carbide. So in 1862, Friendly Waller have calcium carbide interact with the water and produce the acetylene gas. So why I choose the calcium carbide? Because the calcium carbide is non-flammable and easy to dose and easily handling, but acetylene gas is flammable and we can't easily handle, so it's complicated. This is a general reaction of the calcium carbide. So in previous literature, in 2013, Pang et al. Have published a paper by the palladium acetate and uses a bromostyrene and the formation of the annulated product. And also in 2017, Selvaraju have published a paper by the ruthenium catalyzed ruthenium dimer with uses as the styrene and the formation of the annulated product. But in 2021, Liu copper catalyzed construction of the benzo 4,5-imidazole 2,1-isoquinoline using a calcium carbide as a solid alkyne source. So the formation of the non-reduced product.
Excuse me,
So
I have 1 minute left. Please conclude.
Yes, I have synthesized my starting material from 3 methods. So after the studying previous literature, our contribution is the ruthenium catalyzed, where the ortho-CH activation of the 2-phenylbenzimidazole uses a calcium carbide as a acetylene source, and the formation of the two products is non-reduced and reduced product. I have optimized my condition. So the best condition is when the ruthenium catalyzer uses a sodium acetate and the ratio of the product non-reduced and reduced 35 ratio 52. The next is when the increases amount of the ruthenium catalyst, then the reduced product is major product. So this is a general substrate scope of the non-reduced products and the substrate scope of the reduced. So this is a general proximal mechanism, and I have done this mechanism. So I have this is a proton and 13 carbon spectrum of the representative compound. Conclusion, a ruthenium catalyzed ortho-CH activation and annulations were successfully developed using a calcium carbide as an inexpensive and the readily available acetylene surrogate. The protocol enables efficient construction of the annulated heterocycle with good functional group tolerance. Calcium carbide offers the practical and the safer alternative to gaseous acetylene and enhance the operation simplicity. The method demonstrate high the atom economy and align with sustainable CH formulation strategy. So I would like to thank to my supervisor, Dr. Rajkumar Joshi, MNIT Jaipur, and I would also like to the MIT University Gurgaon and all faculty members and my colleagues and family. Thank you.
Thank you, Aarti, for your presentation. So have you ever observed the anagostic interaction for CH activation?
So actually CH activation is the cleavage of the CH bond, presence of the transition metals. The directing group is interact with the metal and provide the near selective site of the cleavage of CH bond.
It could be both anagostic as well as agostic, right?
Agostic, yes. Sir.
So have you ever observed the anagostic interaction for CH activation?
No, sir. Anagogistic interaction, no, because the cyclic intermediate formation through the agostic interaction.
Hello. So what is the distance bond length distance difference in agostic and anagostic interaction?
I don't know, sir.
Okay. Okay.
Okay, sir.
Thank you very much, Arti.
Thank you, sir.
We'd like to call Nidhi Verma next. Nidhi Verma.
Yes, sir.
Are you ready with your presentation?
Ms. Nidhi Verma, please present. You're currently muted. Please unmute and start your presentation.
Please unmute yourself and start the presentation.
Yes, sir. Good afternoon, all. I am audible or not?
You're audible. Your time starts now.
Okay. Myself, Dr. Nidhi Verma, an assistant professor in ITC Group of Institutions, Haridwar. My work is based on the wastewater treatment by the using biochar of pistachio shell with ZNO nanoparticle. But today, in present scenario, there is a wastewater treatment is a big problem, and the major concern is about the industrial dyes and the microorganism. Among all, the malachite green and crystal violet dyes are predominantly used in the industry, which is the dominant pollutant of the wastewater treatment. So there is a need of an efficient and eco-friendly treatment. Among all the advanced oxidation process, AOP is more convenient to comparatively to other processes. In advanced oxidation process, hydroxyl and superoxide oxygen ion generate and plays a crucial role to degrade the pollutants as well as the pathogens. In advanced oxidation process, semiconductor use a metal nanoparticle as a catalyst. In metal nanoparticles, electron and hole regenerate, which are responsible to generate the hydroxyl and superoxide ion, which degrade the pollutants and the pathogens. Among all metal nanoparticles, zinc oxide are widely used because of the good photocatalyst and antibacterial agent and the eco-friendly. The band gap of ZNO is 3.37. So there is a big band gap. To decrease and to increase the efficiency of this, there is suitable matrix is required. Among all, the biochar is a highly effective carbon material because of it. It acts as an electron shuttle having a specific surface area, rich in oxygen moieties and the porous structure. Biochar shows the van der Waals interaction, pi-pi stacking, hydrogen bonding, dipole-dipole interaction, which make it is a suitable photocatalyst to degrade the pollutants. After the insertion of biochar, it enhances the generation of hydroxyl radical ion.
For this purpose, our team has synthesized the pistachio biochar nanocomposite with ZNO. First of all, the pistachio shell is dried and washed with dry water and turned into the kept into the furnace, turned into the biochar. After that, ZNO nanoparticle synthesis by simple soldier method. And after this, the ZNO nanoparticle and pistachio shell biochar are mixed into 1:1 molar ratio, and the composite are formed. Further, this composite is characterized by XRD. As in the XRD shows the in nanocomposite both as well as pistachio biochar and the ZNO zinc particle. ZNO nanoparticle are in zincite form. These are also evidenced by the SAM-ADX. As the SAM-ADX shows, the carbon density is high in the pistachio shell biochar, but it is declined after the insertion of ZNO. And the oxygen peak are also intense due to the highly oxygen moieties present in the biochar. This is the UV-DRS analysis of the nanocomposite and zinc nanoparticle, and it was found that the band gap reduced and found 3.09. Which
You
is
have 1 minute
very
left.
good
Please
for to the
conclude.
degradation of pollutants. This is the photocatalytic degradation of malachite green dyes, or the nanocomposite are also is very useful to degrade the dyes up to 98% in comparatively to biochar alone and the ZNO nanoparticle. Other hand, we have also degraded the other dye, which is the crystal violet dye, and it degrades lesser than the malachite green, but it is degraded almost 50%. It decreases less because of the malachite green absorbed densely, but the crystal violet dye degrade aromatic moiety first. After that, the degradation took place. This is the percentage degradation graph for malachite green, which shows the 98% degradation of malachite green and 50%, almost 50% crystal violet dye. The kinetic study shows that
Ms.
the
Verma, your time is
CO
up. Please stop.
Okay, thank you.
Thank you, Dr. Nidhi, for presentation. So I am just curious to know if you look at your photocatalyst, that is zinc oxide, right?
Yes, sir.
So zinc have D10 electronic configuration. So technically,
Sorry,
which
sir?
electrons are being involved in the photocatalytic process?
Sir, after that, when the light incident, the ZNO electrons goes to the conduction band. Hello.
Hello. So the question
Hello.
is, which electron are being involved in the photocatalytic process?
Sir, which are in the semiconductor ZNO valence band?
No, but zinc have a D10 electronic configuration, zinc +2, right?
Yes, sir. Okay, you want to ask that the d orbital or s orbital?
Yeah, which electron, D or S?
Sir, it is, I think so, in my knowledge, it is a form d orbital.
Thank you, Nidhi. Thank you.
Thank you. Next, we have Wasim Akhtar. Mr. Wasim Akhtar, are you there? Next, we have Khushi Jain.
Hello. Hello. Am I audible? Hello.
I'm sorry, Khushbu, yes.
Hello.
Yes, you're audible. Please start with your presentation. And Mr. Wasim, you can present next.
Okay. Just my screen visible?
No, you're not presenting to us right now.
Ma'am, due to some problem, technical problem.
Hello, ma'am. Is this visible now?
No, Khushi, you're not visible.
Ma'am, I am Khushbu. I think.
Kushbu, please click present, share screen.
I'm sharing the screen, sir, but I'm not able to do that.
Okay, then if Wasim Akhtar is there, please could you present now, and Kushbu be ready to present next.
Sir, is it visible now?
Yes,
I
you're
think it's
visible now.
okay. Okay. A very good afternoon to one and all. Today, I, Khushbu Jain, a research scholar in the Department of CBFS, Amity University, Gurugram, going to present my presentation on the topic cerium oxide, a photocatalyst for ibuprofen and MB degradation. So these are the contents of my presentation. First, I will briefly discuss the synthesis, then few characterizations, then application, and then resultant discussion. So this is the method by which we synthesize the cerium oxide nanoparticles. We used TME seeds. TME is basically HYN. So we reflux the TME seed in water, and the filtered we obtained were used. Its aqueous extract was treated with ceric ammonium nitrate to form the cerium oxide nanoparticles by subjecting it to the precipitation, centrifugation, drying in oven, and calcination. Then the characterization which we performed here. Next. These are the characterization. This is the FTIR spectra, and this is the XRD graph. In the FTIR spectra, we got a dominant peak around 500, which corresponds to the cerium oxygen bond. And the XRD spectra, we obtained for the nanoparticle so synthesized corresponds to the JCPDS 34, which is for the cerium oxide nanoparticles. And this is a UV-DRS study of the synthesized nanoparticles. So in the UV DRS spectra, we got the band gap of around 2.9 electron volt by using the Kubelka-Munk function. And these are the FESAM analysis of the cerium oxide nanoparticles. Here we can see that there are some cauliflower kind of structures which we obtained here. And this is the EDX analysis. In the EDX analysis, the weight percentage and the atomic percentage corresponds to the very well with the atomic formula of the nanoparticles. These are the applications. This is the application. We degraded methylene butyrate at these concentrations, and the degradation of the ibuprofen here. And the degradation was found to be 97% in case of MB, and it was around 63% in case of ibuprofen. So we tried to analyze why this happened, why MB is degraded to a much more extent than the MB. So this is the structure of the ibuprofen, and this is the methylene butyrate. From here, we can see that ibuprofen mainly exists as a negatively charged moiety in the aqueous solution, and MB exists as a positively charged dye, which is a cationic dye. So from the zeta potential curve of the synthesized nanoparticles, it is clear that the zeta potential of the cerium oxide nanoparticles is around minus 49. So as the charge on the nanoparticles is negative on itself, and the charge on the MB dye is positive. So it is more attracted towards the catalyst as compared to ibuprofen, which is basically negative. So here we have shown this structure. Here we can see that cerium oxide attracts more MB than Ibu. So the extent of degradation was more in case of MB as compared to your ibuprofen. So these are the results. We synthesized the nanoparticles, then we did the characterization, then some degradation study, and finally the role of zeta potential we started. Thank you.
Thank you, Khushbu, for presentation. So have you analyzed the product after degradation?
No, sir. No, sir.
Okay.
Actually, we did literature survey from where there we can we were able to analyze.
Okay.
Analyze that.
So is there any reason why you selected that seed for the stabilization of Syrian nanoparticle?
So there are some phytochemicals like thymol in the seeds, and after that we are studying on those work that is active ingredient involved in that. It has many capping and stabilizing agents, which we call phytochemicals.
So most of the plant do have the same, right?
Yes, sir. We tried with the Sadabahar plant as well, but we didn't get the satisfactory result. We are trying to find another way also.
Okay, okay, Pushpa. Thank you.
Thank you very much. Wasim Akhtar, if you wouldn't mind presenting right now. Wasim, are you ready to present?
Ma'am, due to some technical problem, I'm not connected to my file.
Can you stop presenting and connect through so we can call the next presenter? Yeah, you can take your time. Just stop sharing your screen.
Thank you. Deepika Dubey, please.
I'm going to share my screen. Let me know once it is visible.
Saroj Kumar, would you be ready to present right now, please? Saroj Kumar.
I'm sorry, I can go after Saroj because there is some issue in sharing the screen.
Sure.
Present after one of the presenters.
Saroj, please share your screen. Saroj, are you able to share your screen? Can you hear us? Wasim, please unmute yourself.
Mr. Wasim, we're unable to hear you. Can you please unmute and then start with your presentation? Mr. Wasim, you are inaudible, so please unmute yourself and then present.
Good afternoon, respected chairpersons, professors, and participants. Am I audible, ma'am?
Yes, you're audible. Please continue.
My name is Wasim Akhtar. Today I am presenting my research work, comprehensive analysis of heavy metals and dye residue in textile effluent. The introduction part of my research is textile industry is a major source of water pollution, contain dye, heavy metals, chemical, and high complex wastewater. My study area is Bhilwara, Salampura Industrial Cluster, or major textile hub of Rajasthan. High effluent discharge release in this site. And the objective of my study is chemical profiling of effluent and heavy metal analysis, dye characterization, and risk assessment. The simple methodology used in this my research paper. Then first is sampling. The four samplings collected in samples collected in these sites, and ICP-MS for metal, and UV visible, and HPLC-MS for dyes. And the results show very high pollution level. The COD was 920, and the BOD was 310. This is very permissible limit, and heavy metals are observed in this effluent water, and the heavy metals observed in this case, then chromium WHO limits is more than 160 times, and dye analysis also detected the UV visible analysis, and health risk assessment also high, and higher no safe limit in this case, and also significant carcinogenic risk due to chromium, cadmium, and lead. So the conclusion of my research is environmental pollution by textile effluent, and advanced treatment method or strict regulation are urgently needed to control this problem. Thank you, ma'am. Thank you for your attention. I will be happy to answer any questions.
Thank you, Wasim, for presentation. Can you please tell us the significance of this work?
The significance of this problem is it provides the evidence of policymakers and industrial to improve the water treatment and enforce environmental regulations.
So what kind of method are available in market for the water treatment at present?
I appreciate, sir, your question, but based on my current study, I would say, however, this can be explored further in future research.
Okay, Wasimji, thanks.
Thank you.
Thank you very much. Would like to call Saroj to present, please. Saroj, please share your screen. Saroj, am I audible?
Hey, I'm sorry. Can I go next if Saroj is not available? It's Deepika.
Yes, go ahead.
Thank you. I'm going to share my screen. Let me know once you're able to see it.
Yeah, we're able to see your screen. Please start your presentation.
Yeah. So good afternoon, everyone. I'm Deepika Dubey, and today I'll be presenting my work on detecting forgery in scan documents using sensor pattern noise and statistical analysis. So let me quickly walk through what this presentation will include. So this will briefly cover the problems, what existing research says, and methodology we propose to tackle with the digital forgery and challenges involved in the same. So to begin with, we need to understand the context in which this problem exists. So today, most of the documents exist in digital form, but a large number of them are still scanned from physical to digital documents using scanners and or mobile cameras. But the commercial scanners and smartphone captures documents very differently in terms of lightning, resolution, and consistency. That means the same document can look very different if that is scanned using a commercial scanner and a mobile phone. And when you combine this variability with intentional manipulation, there is a huge problem of digital forgery, because there are a lot of free tools that are easily available on internet that could be used to forge a document, modify a document, and make it as you want it. So naturally, the researchers have to go ahead and try solve this problem. And if we talk about looking at the existing, I'm skipping the type of forgery parts and going to literature review directly. So if we look about the existing research, it has evolved from simple pixel-based techniques to more advanced AI-based techniques right now, the current examiners that are, or the researchers who are exploring this. But despite this progress, there is a clear gap that most of these techniques struggle with scanned documents, especially if it is scanned or clicked using a mobile phone camera. So when forgery is subtle and specifically if the document is converted from a physical to digital format, it has become the techniques that are already being defined by various researchers and scientists are not going to work on those type of documents. So to add this gap, we have proposed and we have created this new model where we can combine the sensor pattern noise and block-based patch level analysis to identify the exact regions where forgery has been done. So we propose this framework where the scan document is pre-processed to reduce the noise and normalize the lightning. And then we extract the sensor pattern noise, which is essentially a unique noise signature. We can also call it a device fingerprint. Next, we divide the documents into smaller patches and analyze them individually. By comparing these patches that we have extracted, we can identify the inconsistencies, especially within the certain regions that do not match with the overall document as expected that device fingerprint should be. So finally, we generate a detection map, as you can see in the fifth part, and from where that will highlight the exact regions that were forged. So instead of just looking at the pixels that were done previously by some of the researchers, we combined device fingerprints with localized analysis along with the block-based patch levels to make it possible to detect even a very minute level of forgeries. So what are the challenges that we generally face in such type of documents, especially the documents that are captured using the mobile phones, is that the documents can be edited and rescanned, which makes the very small details missing and the metadata misses. So that is why we are not able to identify as a forensic examiner the exact areas and the regions which were forged. And also there is a huge variation in forgery techniques. So one single method cannot work on different types of forgeries that are currently there in the market. And the metadata can also be manipulated, which is also one of the important points to understand and find out which type of forgery or which type of device was used to capture that particular document.
Amber,
And
1 minute, please conclude in that.
yes, no common data set is also not available currently, and the tools
produces inconsistent results due to lack of the standardization. So this is the result and discussion. In this results, we can see that the model is able to detect the localized tampered regions effectively, even the subtle changes, if we talk about, and the patch-based approach helps us pinpoint exactly where the manipulation has occurred. And this leads us to the important discussion that we observed that combining SPN, that is sensor pattern noise, with patch analysis significantly improves the detection reliability. However, the performance still depends on the image quality, which remains as an important factor, but the digital forgery can be detected with this combination effectively than the techniques that were previously available. So overall, this is a very brief description of my work. If there are any questions, I'd be happy to take.
Thank you, Deepika, for presentation.
Hello. Yeah.
So what is the current stage of your work?
So the work is already completed. So this is just one part of the work, but currently I am using AI, cross-model based AI to detect the forgery using the similar principle that is explained here.
So at what kind of document this research is applicable?
Sir, it is only for the scanned documents as of now.
Only for the scanned documents?
Right.
Okay, okay. Thank you.
Thank you, sir. Thank you, everyone.
Thank you. Can Saroj Kumar present now? Saroj Kumar, are you able to present right now? Okay, up next we have Bhavna Dahiya.
Yes, ma'am.
Please present.
Ma'am, is the slides visible?
Yes, you're visible. You can start.
Okay, very good afternoon, everyone. I am Bhavna Dahiya, PhD Research Scholar at School of Behavioral Sciences and Forensic Investigations at Rashtriya Raksha University. Today, I am presenting my research study titled Establishing the Authorship Through Cross-Script Diacritic Comparison, a forensic study of Devanagari and Gurmukhi under the supervision of Professor Dr. J.R. Gore, lifetime professor at Rashtriya Raksha University. So introduction of the study is like a number of languages has been spoken and written across the world. Likewise, in India, India is also a linguistically diverse country. And a number of languages and dialect, I can say, has been spoken and written. And in India, the constitution has recognized 22 languages in its 8th schedule. And in that, only Hindi and Punjabi is also recognized. So Hindi follows the Devanagari script, whereas the Punjabi follows the Gurmukhi script. Both these scripts are the descendants of our ancient Brahmi script. So if I talk about the handwriting, it is an acquired skill that has been acquired over the passage of period, and it is subconscious writing movement that is basically become the habit for the writing identification. So a number of parameters that basically be utilized for the identification or for establishing the author's identity, that is class and individual characteristics. And from that only, diacritics is a small yet significant characteristic feature that is often habitual and consistent across the different scripts. When a person writes more than one scripts, then there may be a possibility, or there is a possibility that these habits may appear across the different scripts also. So this study basically examines how diacritics in Devanagari and Gurmukhi serves a stable feature for the identification and will act a useful indicator for the writer's specific consistency. So the number of studies has already been conducted using the class and the individual characteristics to establish, to identify the author or the writer, or a comparative study usually like Hindi, English, Punjabi, or from the same scripts also like Hindi, Marathi, and Sanskrit. And a number of studies even from the different geographical regions has also been conducted like Bihar, MP, and Punjab.
Bhavna, can you please share your work?
Yeah, yes, sure, sir.
Please.
So I have basically collected the handwriting samples from the bilingual scriptal individuals from both the scripts, like from Hindi, standard paragraph has been taken and that is converted into Punjabi only across the 3 different age groups. First age group, I've taken like 18 to 30 years, and the second is above 30 and below 50 years, and the third one is above 50. So the standard handwriting samples has been taken. They were asked to write on a A4 size paper, and these samples are scanned at a 600 dpi and later analyzed using a suitable software. So for the particular study, this is only a small part of my study. I have taken the 4 basic parameters that is similar to in Hindi and Punjabi. That is A, I, E, Matra and Chandrabindu. So what basically I did, basically the dimensions of the diacritics I have measured using the software, and I've applied the t-test on that. Particularly 2 sample assuming the unequal variance t-test has been applied. And from the study, it has been identified from the results. Basically, the table shows that in a particular group, like in 18 to 30 individual, it has been observed that A matra shows a significance differences, because this table is basically from the t calculated and t tabulated shows the p value. That is that significant differences were observed at the 5% level of significance. So the A matra shows that p value is much lesser than 0.05, and it shows that it has a highly significant difference. So we are unable to compare based on that matra, and along with that, in that age group only, Chandrabindu also shows the highly significant differences has been shown between the both the scripts. Whereas the E and I matra shows a consistency in both the scripts and has a p-value that is much greater than 0.05, and no significant differences has been observed. Similar to that, only in the age group that belongs to above 30 and below 50, A matra still remains the
Excuse me. Sorry, your time is up now.
okay, just significant differences has been observed in A matra, and the rest matras show no significant difference. And similar to that, above 50, also A matra will remain that it is not comparable and shows the high significant differences. So the study basically demonstrate
Time is
that.
up. Please stop now, please. Thank you.
So what is the significance of this work?
So the significance of this work is basically there is a cases sometimes we receive the questioned and admitted samples in the different scripts. As there is a we have a principle that like can be compared with the likes, and if you're receiving the different scripts and we are unable to compare, but if both the scripts bears the matra, like if we have like Punjabi and Hindi both bears the matras, so we can compare the document based on the diacritics only.
Okay, thank you, Bhavna.
Thank you.
Saroj, are you ready to present? Saroj, this is the last call for you. Are you ready to present right now? Okay, we'll continue. Miss Aditi, please could you present now?
ये देखिए इसको जरा हमारी स्लाइड ये वाली शेयर करो, तो अनम्यूट करो। ओके, आपको अनम्यूट कर देता हूं। आई एम ऑडिबल सर?
Yes, you are. Saroj, are you presenting right now, or should we continue?
Saroj, please take your time. We will move on to Aditi Chauhan.
Hello, good afternoon, everyone. My name is Aditi. Am I audible?
You are audible. Kindly present your screen as well.
Okay, sure. Just a second.
You may start presenting now. Your time has started.
Ma'am, can you see my screen?
Yes, you can present now.
Okay. Good afternoon, everyone. My name is Aditi Chauhan, and the topic of my presentation is comparative analysis of quencher and conventional solid phase extraction for extracting benzodiazepines from human blood. So this is the content of my study. Benzodiazepines are pharmaceutically available synthetic drug which causes mild to severe depression. They cause depression by increasing the action of GABA in the brain. These are the drugs which I have used in my study. These are clorazepam, diazepam, and etizolum. These are available in pharmacy after the prescription. Aim of the study. So long-term use of anti-anxiety results in addiction, which leads to anxiolytic abuse. This misuse of anxiolytics leads to overdosage, which sometimes results in poisoning. So the case of poisoning deaths become a matter of concern for forensic toxicologists to develop a modified, rapid, and environmental friendly extraction technique. No doubt we have so many extraction techniques, but such as liquid extraction, solid phase extraction, but I have tried to modify a new extraction method, that is quencher, which uses less solvent, so we can say is a environmental friendly method than the conventional methods. So the objective of my study was to compare the concentration of drugs in biological specimen like blood using modified quencher technique and conventional SPE extraction technique. Experimental strategies. So I have taken 10 healthy individuals in 10 healthy individuals, and I have taken the blood sample. 5 ml blood was taken, and these were spiked with clorazepam, diazepam, and etizolum drug. So the quencher extraction technique and cartridge solid phase extraction technique. So for quencher extraction, I have taken 5 ml of blood sample, and spiked it with different concentration of 5, 10, 20, 50 ppb of the drugs, and these were vortex. And then I have added methanol as a organic solvent, and quencher salt pouch has MgSO4 NaCl, through which the phase separation occur, and organic phase was the upper solvent layer was transferred to the other vial, where the cleanup phase was done using DSPA 500 mg magnesium sulfate and 250 mg PSA to remove the organic content from the solvent. So then it is vortex, and we have transferred the 10 microliter sample to the LC-MS for further analysis. Whereas in cartridge solid phase extraction, I have taken the cartridge SPE cartridge was used, which was conditioned with 3 ml methanol and 3 ml deionized water. Sample was loaded, and the elution was done using ethyl acetate, which containing 2% ammonium hydroxide. So these were the procedure which I have followed for both the extraction technique, and the method development for modified quencher was
One minute
optimization.
left. Please conclude on that.
Okay. So I have compared the recovery of both the methods and found out that both the methods falls between 70 to 120% range of the recovery, which means both the methods were ideal to be used in forensic science laboratories. You can see the recovery percentage of conventional solid phase extraction and the DSPE, that is modified quencher extraction technique. Then second was the matrix effect. Matrix effect was compared by comparing benzodiazepine in neat solution, that was without matrix, and one is in the matrix, and we found out the relative matrix effect was less than 20%. All the ranges were between 4, 3, 3. So the method was adequate. Next was a precision. Basically, 6 different replicates were used for the same analysis, and we found out that our
Betty, your
percentage.
time is up.
Okay, so this is the conclusion that both the extraction techniques are good, but since modified extraction uses less solvent, so we can jump into modified quencher for our further investigations in forensic science lab.
Thank you,
Thank
Aditi. Okay.
Thank
Thank you, Aditi, for presentation. So I have just one query: how you can extract 120% out of
Okay,
a drug?
sir. Sometime what happens with the main principal analyte, we get contamination also. So after 100% is like you have you have got all the 100% analyte. But with the analyte, if we get contamination and our this thing is increased, so the basic European 808 guidelines of validation is stating that 70 to 120% is the acceptable range. Like if the contaminations are also recovered along with the main analyte. So only 120% is acceptable.
Okay, okay. Thank you, Aditi.
Thank you so much, sir.
Thank you, Aditi. We would like to call Saroj Kumar
Yes,
one last
yes,
time.
sir. I am audible, sir.
Yes, you are.
Yes, sir. Today I am presenting. Sir,
Can you stop sharing?
my slide are visible.
Yes, you are. You're visible, Saroj. Thank you.
Yes,
You start.
yes, sir. Thank you, sir. My name is Saroj Kumar. I am research scholar, University of Lucknow, Department of Chemistry. Today I am presenting here my work, profiling of primary and secondary metabolite in the different variety of millet with its nutritional benefits. These are, as you all know, millet are rich in the nutritional profiling in the all the crop are used in the, mostly in the our nutritional component. These grain are known for their nutritional and therapeutic uses, or they are short duration of crop, 60 to 90 days, can grow under low water input condition. Input means that low fertilizer and poor farmer can be easily affordable of these crop cultivation. They provide food as well as nutritional security to people living in semi-arid region because of adaptation wide range of agroclimatic ecosystem. Here we are showing you how the metabolite are necessary for our healthy life, because after the deficiency of metabolite, many diseases are caused by the deficiency of these necessary metabolite. So millets are the rich in the various metabolites such as primary and secondary metabolite. Primary, that is means that which are directly involved in the body, directly physically support in the body development, and secondary metabolite that are the phenolic, flavonoid, which are also known as phytochemical.
Saroj, can you go directly to your work?
Ji?
Can you directly go to your work, what you have done?
Yes, sir. Yes, sir.
Right, please.
Here we are comparing the nutritional profiling of the 7 variety of millets that are mostly cultivated in Uttar Pradesh, India. These here we are showing you that the most important millet which are rich in the protein, that is proso millet, and second one is the foxtail millet. Proso millet rich in the protein, but foxtail millet have rich in fiber content. So millet are more sufficient for the diabetic person, diabetic patient. Here we are comparing secondary metabolite in the same variety of the millet, that is that are included in
There's
the
1
same.
minute left. Please conclude.
Yes, sir. Here we are comparing the flavonoid content in total 7 variety of millets, and overall these are the HPLC profiling of the millet, and finding that catechin and protocatechin are the rich in the protein, rich in the millet crop. Conclusion: millet are rich source of metabolite, including both primary and secondary. Protein content are sufficient to build body muscles and cell regulation. Carbohydrate present in required for energy providing to body. Fiber content are present that protect body from the chronic diseases. Millet are rich source of phenolic, flavonoid, antioxidant property. Significant amount of the nutrient are present in the millet. So I need to include in our diet. Thank you, sir.
Thank you, Sarojji, for presentation. So have you used HPLC for the identification of these carbohydrate, protein, vitamin things?
No. I use the HPLC only for the secondary metabolite, that is phenolic content profiling.
Right. So what method have been used for the identification of the carbohydrate, protein, vitamins, and other thing?
Protein basically you estimated through the Lorry method and Bradford method. Carbohydrate to estimated through the Boltz-Williams method. And next, sir, what is the question?
What about the vitamins?
Vitamins, folic acid, or many of the vitamins are present in the millet crop.
What is the accuracy of these results?
Accuracy means that we are taking the data in that biological triplicate manner, but we are not apply here the statistics value.
Okay, Saroji, thank you.
Thank you, sir. Thank you so much.
Thank you very much. Now I would like to call Anupam. Anupam, please share your screen. Anupam, can you hear me? Am I audible to you? Please unmute your mic. Okay, thank you, everyone. That concludes today's session.
You all are free to leave the meeting. Thank you.
Thank you. Anupam, this is the final call out. Can you hear me? Please share your screen, unmute your mic. Okay, thank you very much, guys. You may all leave.
IT team, please end the meeting.
Thank you very much. All participants, meeting is over. Thank you very much.
IT team, please end the meeting. We should exit.
Hello.
Anupam,
Myself, Anupam, audible, ma'am.
yes, you're audible. Please share your screen and
Ji,
start
sir.
your presentation.
Okay, sir. Sir, myself, Anupam,
Please share your screen and
PhD
start your
research scholar
presentation.
under. Ji, sir.
Please share your screen.
सर स्क्रीन नहीं शेयर हो पा रही सर.
Okay, then just do your presentation without the screen. You have 4 minutes starting now.
Okay, sir. Thank you, sir. Myself, Anupam, PhD research scholar under the supervision of Professor Sumitra Maurya, Department of Chemistry, Sri JNM PG College, KKC. My research topic is polyphenolic composition and in vitro antioxidant activity of hibiscus rosa sinensis L, implications for pharmaceutical applications. My first slide is introduction. Polyphenols constitute diverse group of secondary metabolites found in plants known for their antioxidant and anti-inflammatory and anti-cancer properties. Medicinal plants are rich source of polyphenols, making them subject of intense scientific investigation. India stands as major nation boasting 40% of world's biodiversity and a wealth for rare plant species. Within the diverse flora, medicinal and aromatic plants form a significant component supplying essential raw materials for pharmaceuticals and drug industries. India's heritage encompasses ancient medical systems like Ayurveda, Siddha, Unani, and various indigenous healing traditions, all rooted in the utilization of numerous medicinal herbs. A staggering count of more than 9,000 native plants have been documented for their healing attributes. My research work is the extraction yield of hibiscus rosa sinensis and leaves obtained using two different solvents was evaluated. The results showed that ethanolic bark extract of Hibiscus rosa sinensis L yielded at higher percentage compared to the ethyl estate leaf extract, indicating the efficiency of ethanol as extraction solvent.
So you have 1 minute. Please conclude in that.
First, we calculated the total phenolic content. The total phenolic content was estimated using the Folin Calcio method by Singlet and Rossi in 1965. The milli was used as a blank and gallic acid as a equivalent is used as a standard. The result indicated that the highest TPC value for the ethyl estate extract was 22.22 milligram gallic acid per equivalent, whereas the ethanolic extract showed a total phenolic value of 15.37 milligram gallic acid per equivalent. Then we find total flavonoid content. The total flavonoid concentration of each extract was determined
Okay,
using the
Anupam.
aluminum. G. Okay.
Anupam ji,
G.
am I audible? Thank you. Thank
Ji,
you for
sir.
your presentation.
Ji, sir.
Can you please tell us what is the reason why your compounds are more soluble in ethyl acetate than ethanol?
Sir, is this solvent is low polarity.
Ethyl acetate has no polarity?
Ji, sir.
Ethyl acetate is pretty polar.
Then ethyl acetate low polarity other than like water, alcohol, hexane, and petroleum ether.
So ethyl acetate is more polar than hexane or less polar than hexane?
Less polar than hexane.
Okay, okay, Anupam ji, thank you.
Thank you. You may all leave the meeting, and I request the IT team to end the meeting now. Excuse me, ma'am.